Hello everyone. Welcome to this. Well, let me start again since we just started the live stream. So welcome everybody again for this last day of lectures. So it has been a long marathon so far, and we see the end of this nice school, which I really enjoyed. And so today we have the first lecture by Vishwesha Guttal, and then we're going to take a 1 hour and a half break. And then we're going to have one panel, two lectures, and one last panel. So without further waiting, I'd like to leave the floor to Vishu to share his screen and start the presentation. So thank you very much, Vishu, for being here.
Hi everyone. Are you able to see my screen?
Yes.
Okay, excellent.
Is not okay. Yes.
It's now full screen, right?
Perfect.
Okay, great. Thanks everyone again for joining me today. And I know it's the end of the third week of the long workshop. I hope I had great fun, and I know I really appreciate every one of you who has sort of been hanging on and attending the last day of the lectures as well. So since the last day, I will try to sort of go slow and try to be interactive. So if I do not if I'm going if I'm not clear about anything, somehow I don't get any sort of message when I sort of when there is a question over the chat box. So maybe somebody can prompt me, or just unmute yourself and speak. So that's also fine if you have a question that you would like to ask. So today, in the third talk of the series of talks I am giving, I'll talk about how intrinsic noise that is sort of within the system, um, sort of and the bistability interact with one another in very interesting ways in animal collectives. So, of course, in the last talk, I spoke to you about this self-organization patterns in plants. So these are collection of plants, and today I'm going to focus on animals. In plants, what the fundamental principle I sort of mentioned was that there are these feedbacks between plants that cause different types of patterns. For example, this simple example of local positive feedback that decays to zero after a finite distance, that will give you irregular patterns where the cluster sizes are not same. On the other hand, if we had a local positive feedback followed by a short range negative feedback, then this leads to periodic regular patterns, which are also very popularly known as Turing patterns. Okay. So likewise, there are these animal patterns, and I also spoke about bistability and noise. I know this is just to remind you, because I will make some connections to this in the end. So we thought of bistability as a potential well and a ball rolling in this potential well with two minima. And system can go back and forth between two minima. Just like the ball, can bounce back and forth between two minima because of stochasticity. So just to remind you the basic concepts. So, but today I'm going to talk about noise, bistability, and patterns in animal collectives. And to do so, I will start with this video. I hope this plays well, and I hope my network is not too bad. So here you see a sort of whole range of collectives of animals, birds, fish, krills, and many other species. You will also find these kind of patterns in cells, bacteria, where as an aggregate, they're performing extraordinary synchronized motion. And what you're seeing in this specific part of the video is how a simple computational model can reproduce some of these observed patterns. So again, this is actually a simulation. What you're seeing right now is a computer simulation, and how it can fairly accurately describe the observed patterns. So, and here is an example of a collective motion from the laboratory where we work. where we have just placed a whole bunch of fish in a fish tank. So these are about 750 fish in this specific case. Again, without any stimuli, even in the experimental conditions, we actually can observe these kind of collective motion and synchronized movement patterns in animal groups. So the sort of broad outline for my talk today, with that motivation of couple of videos, is to understand how do animals exhibit this collective motion, and what are the basic mathematical and computational models that tries to explain this. Okay, and I'm going to sort of argue that one can use stochasticity as signal in understanding this collective motion. Okay, so I'm going to sort of explain that in steps, mathematical theory behind this, what are the methods and experiments that we have done to sort of show that. And if I have time, I will also talk about some of our ongoing work on dynamics of collective motion under predation. Okay, so when we think of these animals in groups that are moving, people ask sort of two classic questions. One is that of ecological or proximate question. How do organisms exhibit such remarkable collective motion patterns? So to sort of break this question further, what is it that each animal is interacting with other animal, other organism, that produces these patterns? Okay, that's the basic mechanistic question that one is interested in. And secondly, one can also ask, why do organisms exhibit collective motion? What are the costs of being in groups? What are the costs of moving in groups? At the same time, there has to be some benefits that animals gain by being in groups and moving in groups. How does the trade-off between these costs and benefits sort of weigh? So those are two broad class of questions people try to understand in the field of animal behavior. So today I'm going to focus only on the first question. How do organisms exhibit these patterns? More specifically, how is each fish or each bird sort of talking to or interacting with its neighbor and producing these patterns? So here is a sort of a classic model of collective motion. So it's some of the early models of collective motions were developed by computer scientists and physicists in 80s. Those are known as the Boyd models, for example, developed by a computer scientist, Derek Reynolds. They're also widely used in animation industries. And later on in 90s and early 2000s, there were more sophisticated, more detailed models from a physics point of view as well as biological point of view. So here, the model I'm going to present today is from Cousin et al., 2002. So here, basically the idea is we can represent these groups of fish or insects or birds as actually point particles. Although I'm showing you cartoon pictures of fish, you basically assume in the model that these are all point particles, but with some rules. And what are those rules? There is a tendency for animals to exhibit local repulsion, meaning if two fish are too close to each other, they repel from each other in their movement. And secondly, once they're not too close to each other, they exhibit these two social interactions. One is that they have local attraction with each other, and that causes cohesion. And they also have local alignment with each other, that causes a directional alignment of the entire group. And one can write down equations of motion for each fish in these groups. Okay, and then if you sort of iterate these rules over using computer simulation approaches, what you find is that animals may follow these very simple rules like local attraction to stay cohesive and local alignment to stay in the direction of motion of neighbors. They can actually show striking patterns. So here is an example. So depending on this, so I spoke to you about two important forces or interactions, right? Attraction and orientation. For example, if the attraction is very, very strong and the orientation is not very high, you often will find a cohesive sort of group of animals, but they don't have directionality in their movement. On the other hand, if the orientation is the dominating force, if the animals are strongly aligning with each other, then what you find is they not only form groups, they also move in a polarized fashion in some arbitrarily chosen direction. And finally, here is something very interesting that's very hard to guess. If there is some balance of this attraction and orientation, you find patterns like this, very counterintuitive. Why do I say it's counterintuitive? Because there was nothing in these rules of attraction and alignment that tells them to go in circles, right? Yet, if you put them together, if every individual fish, every individual insect follows these rules with some strengths of attraction orientation, you can also find them just going in circles. Okay, so basically these kind of models can not only explain observed patterns of collective motion, they also make predictions about new forms of motion that were not very obvious. And in fact, there are many organisms that show this kind of milling behavior. Okay, so those are the simple, I have just introduced you to one simple class of models. These are also called self-propelled particle models. And in this context, okay, there are all these models. How do we know? How do we know they're right? How do we know what are real organisms doing? Are real organisms following these simple equations of motion? Are they really exhibiting these local alignment, local attraction rules? Uh, but to address this problem is really challenging, right? Because imagine I observe flocks of starlings or schools of fish, hundreds of them, tens of them. By observing them or by recording them, how can I really infer what's happening at the local roots? It's not very obvious, right? And to counter this problem, people have devised experiments with very small numbers. So I'm going to give you a very brief review of this. So this was one of the very first papers trying to understand how animals move in groups. These are what they did. This is a group in Rome, which I'm sure many of you might be familiar with. And what they did was they recorded and they sort of took very high resolution images of starling flocks. And what they did was, from these images that were taken from multiple cameras, they were able to reconstruct the positions and the orientations of each starling in the flock. And from analysis of the positions and a quantity called anisotropic factor. So I'm not going to explain what exactly this is because that's a bit technical. And what they basically conclude are two things. First is that the starlings interact with one another not based on distance, but rather based on who are their some hierarchical neighbors. For example, what they found was they interact with 6 to 7 neighbors independent of how far they are. So the distance doesn't matter, but what really matters is that they are interacting with, on an average, 6 to 7 neighbors. And once you and with that kind of a inference, they were able to sort of reproduce many of the observed patterns in the model, in the data. And there were a whole series of other work where the so I'd see at one extreme here you have large number of starlings that are moving, and from them you infer what they are doing. The one other extreme is that do experiments in the laboratory, put only a few number of fish, 2 fish, 3 fish, maybe 5 fish or 10 fish, and from the movement of those fish, people have tried to infer what are the rules that animals are following. So there are many such approaches. I'm not going to go to a whole review of this, but the sort of summary of many of these studies is that the simple physics-inspired self-propelled particle model that I mentioned are not really what the animals seem to be doing. Animals seem to be doing something sometimes often much simpler, sometimes much more complex. So animals are probably doing something fundamentally different. So nevertheless, there are some challenges in understanding what are the rules at the level of individuals. For example, if you were to do experiments at the level of only 2, 3, or 4 fish, you have inferred how are they interacting when they are in 2, 3, or 4 fish groups, right? However, how do we know they will be true even when I put in 25 fish? Maybe the rules of interaction among fish will change as the group size increases. because the density increases, maybe they have to do something different to stay cohesive and stay polarized in their direction of motion. So it's not very evident whether these rules will scale to larger groups as well. And secondly, many of these studies sort of ignore the role of stochasticity in how interactions happen. So, it's also not very clear whether the inferred rules are unique. For example, the more detailed observation you make, you will find a lot of interesting but detailed behaviors. But the question is, are they important? What do they really tell us? Obviously, there will be differences among fish even within a species, and there will be differences among individuals from one species to other species, and of course naturally across taxa. So it's not very, so one must really aim to understand some basic underlying principles of this collective motion. Okay, and in the context of understanding how animals interact, it also really depends on the scale that we are studying and the context we are studying. For example, if my question is what makes group cohesive, I will get some set of rules that keeps animal groups cohesive. On the other hand, if my question is what makes group motion highly polarized and directional, and that will determine, that will give me a sort of maybe a different set of rules, right? And moreover, the ecological context matters. For example, are these animals facing predation, foraging, and that again might determine the types of interaction animals are following. So in this sort of broader context, the focus of my presentation today is a recent work that we have done, where we try to understand the dynamics of group polarization. The first thing we see when we observe a group of animals moving is how nicely polarized they are, and how sort of spontaneously they change these direction of motion, and there is some element of stochasticity, and there's some element of determinism in them, right? How do we understand these dynamics of polarization? So given that the focus is on polarization, I'm going to focus mostly on deciphering the rule of directional alignment in collective motion. And in doing so, I'm going to hopefully convince you that the role of noise, one must not really ignore noise, and there is very interesting signatures that the noise can provide. So how do we do this? I'm going to first describe the experiments we did. Okay, so what we did was we took a fish tank. With shallow width, and what that really helped us was, because it's a shallow depth water, fish were effectively swimming in 2 dimensions, and therefore we could record all the group members from a camera mounted on the top of the fish of this tank. So here is the picture on the right shows a snapshot where we have observed the fish school is somewhere near the center. They're all moving towards the right part, and then we use techniques of image processing to identify the coordinates of each fish as well as the direction of motion of each fish. And this inset here shows the colors show the trajectories of each fish over a short duration of time. And the fish we are using is called karimeen. It's a fish that is native to southern part of India. Okay, so here is a video, I hope it plays, of how these fish schools look like. I'm showing you a group size of, I think, 60. Okay, you can see how they're mostly cohesive. Occasionally there are some individuals who move away, but on a longer time scale, those are really rare events. And we do these experiments for about 1 hour long, and we have done these experiments for 15 individuals, 30 and 60 individuals. And then as I explained to you, we did what's called image processing, which is basically by the fact that between 2 frames fish have moved some distance, we can construct the direction of motion. And once we have done, we know so on this slide, focus on the top right corner here where I have showing VI hat. So VI hat is the direction of motion of the fish I at time Tn. And if I know the direction of motion of each fish, I take the vectorial average and we get something called a group polarization, M of Tn. So what is this M? If I look at the modulus of this vector, if this vector is 0, the fish are moving in random direction. On the other hand, if the modulus of this vector polarization is close to 1, that means that fish are nearly all aligned with each other and they are moving in the same direction. And you do this analysis for every frame of your video. Actually, we don't do it for every frame. We skip a few frames in between because sometimes fish just don't move because between consecutive frames. So from that, we calculate this time series of polarization as a function of time. We are showing you here only for 6 minutes, but this is roughly, the entire experiment is 45 minutes long, and there are many such replicates. So what you are seeing here is the following. On the top figure, you are seeing the polarization for group size 15. In the bottom two panels, you are seeing polarization for 30 and 60 fish individuals. Visually, you can already observe a whole bunch of patterns. The first one is that the group size N15 seems to be polarized in general more than these two, the higher group sizes. Secondly, there is a lot of stochasticity in the data. The time series is fluctuating fairly rapidly over time. The polarization is rapidly fluctuating over time. Okay, this is the experiment we have, right? But I mentioned to you one of the questions that people have been interested is, okay, we have all this collective motion, that's great, we can now quantify them, that's also fabulous, right? But ecologists and often want to understand what is it that each fish is doing, what is each bird doing. Can you tell me what are the individual level rules that produces these patterns? Okay, so how can we derive these rules from the noisy data that we have? Okay, so to do that, I have to explain some theory to sort of, we have this data, how do we analyze this data? For that, I have to now go back to some theory. So what's the theory? So I'm not going to go into a lot of mathematical details. I'm going to hide the mathematical details. Okay, so what I am going to show you is the following. Look at these two graphs, one on the left side, one on the right side. What are you seeing? You're seeing y-axis polarization. Okay, before I go, are there any questions at this stage? I can take any questions if something was not clear about the motivation or anything about what I've said so far. There is one question in the chat box, or no?
No, there is no question. It was me.
Okay. Okay, no problem. Yeah, before I go further, I can take any questions if there are any.
Yeah, if anyone wants to raise a hand, let me check.
No, so I can probably go forward.
Hi, sorry, I have a question.
Sure.
I apologize because I didn't follow last lesson. I couldn't be there. So maybe my question is redundant. By the way, like when you take in considerations like this kind of behavioral models, like of fishes moving, sorry for my words, do you take in consideration also the environment in this modeling, like let's say current, or your aim is the one to isolate only the motion of the group?
Yeah, that's a great question. So in this part of my talk, I'm going to assume that the environment plays no important role. So you're right that imagine if I was studying bacterial motion in their medium, the environment actually is really important. The viscosity of the fluid will be very important, right? Whereas when fish are moving in water, often it's not very important. The self propulsion is sufficiently strong, okay, that maybe the fluid dynamical effects can be ignored. Okay, so but I could be easily wrong because where I'm not aware of there is only one recent study that looks at 2 or 3 fish and a fluid dynamical effect. I haven't read that very carefully, but I'm not aware of any study that has quantified this effect as of today. But that's a great question.
Thank
As
you
of
very
now,
much.
I'm going to ignore that. Okay.
Thank
When you
you.
say environment, there could be other aspects of environment, right? Imagine
Of course.
there's a predator, right? That's also environment for the fish. It's an ecological context. That also plays a role on how they move. That is something, if I have time, I will cover in the second part of my talk.
Thank you very much. Cheers.
Okay. So, okay. Any other, if there are no other questions, I will move forward now. So, okay. So how do we analyze these data? I have computed this polarization as a function of time, right? How do I analyze these data now? Okay, that's the basic idea. So now I'm showing you 2 graphs here. What I have done here is I have plotted the polarization as a function of time. And as you will see, the polarization is varying between -1 to +1. Ignore the fact that there is a negative sign here. Trust me that that's not very important. But what you are seeing here is that these 2 graphs are pretty similar qualitatively. But but how did I get this data? I actually got this data from computer simulations of 2 very different types of models. Okay. But they look very similar, right? They are fluctuating between values of +1 to -1, right? And there is a lot of stochasticity. And if you plot the histogram or maybe the normalized density functions for the polarization, you find it's bimodal, meaning the value of m is either at +1 or -1. Okay, by the way, let me just tell you what is +1 and -1. What we were doing in the simulation was we were assuming that the world is a torus and animals can move only in one direction or the other direction. So if everybody moved in the direction, let's say counterclockwise, the map polarization will be +1. If everybody moved in the counterclockwise, the polarization would have been -1. Okay, so that's what I'm plotting here. Okay, so the histogram of these 2 look pretty similar. Of course, they're not identical. There are some differences, but the differences could be entirely quantitative. There's nothing fundamentally different between the 2, right? Okay, in fact, let me now tell you what was different between these 2. So the left column, I produced these values of polarization, assuming that each fish in the model interacts with only one other fish and copies the direction of motion. And then every fish keeps on doing this. That's how I produced this left column data. On the other hand, in the right column, I assume that each fish looks at 2 other fish and then it moves in the direction of the average of the other 2. Okay, approximately speaking. So as you're seeing in the left column, there is only a pairwise interaction. One fish looks at one other fish. There's a pairwise interaction. On the right column, the fish, there is a ternary and a triadic interactions. There are 3 fish and they all move in the direction which is the average, which is the average direction. They both produce very similar time series of polarization and they both produce similar PDFs. How do I know if I only had time series? How do I know where it came from, right? So to address this question, let me now describe the model in more detail and I'll tell you some results from the model. So what is this pairwise interaction model I just showed you? In this pairwise interaction, imagine there are a large number of fish, okay, and then you just pick 2 fish randomly. So you pick basically a pair, arbitrarily chosen pair, and then one of the individual will turn towards other at a rate c. Okay, so this is a probabilistic interaction happening at a rate c, and one of the fish will turn towards the other at rate c. So as you can see, after this copying interaction, both the fish are moving in same direction. In this, there's an additional rule because we want to have some randomness in the system, which makes sense. A fish turns randomly by angle delta theta at a rate s. Okay, so there are basically a stochastic turning and a turning caused by copying a randomly chosen individual. This is what we call pairwise interaction model. Now I will discuss what is called the ternary interaction model, the right column of my simulation results. So in addition to pairwise and stochastic, we have those two. We continue to have them. We have now added a new rule called ternary or average interaction. So now what is this? How do we implement this? Look at this. There are three fish at the bottom here, right? On the left side of the bottom, where my mouse is moving. So this fish, which is turning away from the slide, is most disoriented in this triad. So what this guy will now do is turn towards the other two. So for example, the result of this ternary interaction is that this fish who was moving away from the slide is now moving towards the two other fish. So in some sense, this is causing an average effect. And that's why we also call it average interaction model. So unlike the pairwise interaction here, there are three fish involved in an interaction. And this happens at a rate h, which unfortunately I forgot to write. So the rate of interaction is h here. So I have described two models now. So what do we do with these two models? What we do is, we construct analytical model, and we try to look at the same quantity I showed you in the empirical world. In the empirical world, what did we do? I calculated this capital M, and I'm denoting here by small m for the models. The small m here is basically the summation of the velocity vectors of all the fish. Okay, so it's basically the average of the velocity vectors of all the fish. Okay, so we now want to understand what is the dynamic of this quantity m. Now, I'm not going to show you the mathematics. So it involves, as you can see, the rules were stochastic, right? And what we basically do is use ideas of master equation and something called system size expansion in the physics literature. And from that, what we do is we calculate what is a stochastic differential equation that describes this group polarization. Okay, so all the mathematical details are present in this citation. It's also available on the archive, so the physics archive. So you don't have to really buy this book. So what we find is the following: if the pairwise interaction model is correct, the dynamics of this m follows a stochastic differential equation given by this. So I know there are lots of terms here. Let's and then if the ternary interaction model, we find another equation which is again SDE, which is given by this. Now, what should you focus on? This there are so many terms, right? Let's focus on the deterministic term. Okay, so even before that, let me point out that there is a stochastic term eta t here on the right hand side. Okay, on eta t is basically the Gaussian white noise. That's a stochastic term. And then we also have a deterministic term, which I have circled here. Now let me just try to see if okay. So we are focusing on this deterministic term. Clearly, you see that the deterministic term in the pairwise model and in the ternary interaction model are different. The pairwise interaction model has a simple linear term AM, right? Whereas the ternary interaction model, in addition to the AM term, also has a cubic term. Okay, and this H is the ternary interaction rate, which was not present in the pairwise interaction model. Therefore, basically, if you can compare the deterministic part, if you can somehow compute the deterministic part of your data, then you can distinguish whether your fish are following a pairwise interaction model or a ternary interaction model. On the other hand, you can also look at the stochastic term. Okay, the stochastic term again has a similar form. They are both quadratic in M. However, looking at the stochastic form here alone is insufficient because they are both quadratic. So one has to look at a combination of deterministic and stochastic term to distinguish these two models. Okay, so that brings me to the following question. We had, okay, before I go further, I want to say something. Okay, something about bistability and stochasticity. Okay, So you remember that in the previous talks on tipping points, I discussed this classic picture of a ball in a potential well. I think a lot of things moved. Oh, I don't know how it moved. Okay. Oh, it's somehow overly sensitive. I don't know. I think I'm not using this correctly. Just give me 1 second, please. I'll try to erase this first. Oh, something happened. I don't know what happened. Okay, I think it's fine now, but I was hoping I can write, but there is some problem. Okay, we remember we discussed this picture of ball rolling in a potential, right? If there are two wells in a potential, because of stochasticity, ball can move back and forth. And then if you now plot the time series, you find that system will go back and forth. Okay. On the other hand, imagine another system where you have a single well. The ball is typically, what do you expect if there is stochasticity? What do you expect? That it just fluctuates around this mean value, right? That's what you typically expect. However, whenever we say this, we often make an assumption that the stochasticity is constant for all values of the state variable. Let's go back to the previous slide. Look at this. There is a deterministic term, which is simply a exponential decay. There is a stochastic term, which is not a constant term, which actually has m dependence. So let me write down the equation again. So what we really have, so typically when we study many systems under a linear approximation, we do something like this. There is a exponential decay of the system from many perturbations. Therefore, m is equal to 0 is the fixed point. And then you have a stochasticity that makes a system go around the equilibrium value. What we have in the pairwise interaction model is something very interesting. What we have is this stochastic term is actually not a constant term. It actually has a m square dependence. What does that mean? What this means, when m is equal to 0, deterministic term is 0. However, the stochastic term is actually maximum, a plus 1 in this case, the way I have written it down. Imagine when m is now close to 1, if m is 0, right? The stochastic term is basically a plus 1, the way I have written. On the other hand, imagine m is equal to 1, which is the maximum order parameter, right? Maximum value of the order you can have. Then the stochastic term will be actually minimum. So meaning when the system is disordered, which is m is equal to 0, we have least amount of stochasticity. When the system is ordered, we actually have lowest amount of stochasticity. So this noise is playing a very interesting role here, because when the system is highly disordered, the noise is very high, and therefore what the noise then does is it actually kicks the system out of the zero equilibrium and produces a pattern like this. Okay, so the pairwise interaction model basically has a fairly counterintuitive effect of stochasticity. So it's also okay. So with that description, let me now come back to the fact that we have this two time series now. One of them came from pairwise copying, the other came from ternary interaction, and they both look similar. So if we can somehow construct a stochastic differential equation for these data sets, then I can distinguish where the data came from. So how do we do that? So there is indeed a method that actually can be used to construct where the data came from, and the basic idea is as follows. So the deterministic term basically is the average change in the value of the order parameter. On the other hand, the stochastic term is basically more like at the second moment of the change. Okay, so basically by computing the first moment of the change, it's called jump moments. First jump moment will give me the deterministic term of the SDE, and the second jump moment will give me the diffusion or the stochastic term in the stochastic differential equation. Okay, and in fact, if you apply this method, here is what you find. Here on the right hand side, now focus on the right hand side. If I use the data from the pairwise copying model, if I use the time series, and if I apply this formula, I do actually find the deterministic term is a linear term. On the other hand, if I apply the method to a ternary interaction model, the black solid line is the answer here, by the way. We find that, in fact, this term is actually cubic. And by comparing the fact that these two functional forms are so fundamentally different, we can infer that this data therefore came from ternary interaction, whereas this data came from the pairwise interaction. Okay, so that's the basic idea on theory. I will now stop for a few seconds and let you ask any questions, because some of these theoretical concepts may not have been well explained.
So if there is any question, there is no question in the chat, but
okay. There are no questions. Okay, fine. So I will go ahead and okay, there is one question.
There is a question from Tuan who is asking, it seems a kind of kinetic Ising model, right?
Which one of them is a kinetic Ising model? This is really closest to so I think the pairwise model is more like a water model. Okay, the ternary interaction model is more like a kinetic Ising model, because in Ising model you basically have the Jij sort of, you look at the not just one individual, one spin and turn, right? We actually often do a average of the neighborhood of the spins. So that's more like the ternary. That's my understanding. But in some limit, you're right. They're actually more like XY model, because you observe if you think of it in 2 dimensions. So basically, whatever I showed you here was all in 2 dimensions. All the vectors were in 2 dimensions. So all the analytical results are also from 2-dimensional. So all these all these M vectors are actually 2-dimensional vectors. So they're really more like XY, kinetic XY model with different interaction rules.
There is another question from Rajwal, who is asking, the criteria to distinguish between interaction rules come from top-down approach. How well does the criteria apply to bottom-up approach, where you start off with observational data and then try to plot the jump moments? Yeah,
that's a great question. And I think I have clearly done a very poor job of explaining this slide, which I have currently, I'm showing now. So the point of this slide is actually exactly that, which is, sorry, I think my slides went off. Okay, so that's precisely the point of this slide. I showed you, if I have a model, I showed you how to get the equations, right? What this slide is showing, and again, I forgot to cite the paper here. We have a paper early this year that shows this. What we have shown in this paper is the following. If you gave me only this time series data, which could be observational data or simulational data, here I am demonstrating for the simulational data. If you had given me these 2 time series without the labels of pairwise and copying, I would then apply this formula of capital FM, and then I can construct the deterministic term for both of them. And then by comparing deterministic term, I can conclude that the pairwise model, the left column came from pairwise because it's a linear term. The right column data came from a ternary interaction model because it's the data, because the function is cubic. Did I explain that point? So this is the bottom-up approach. So given data, how do you construct the jump moments? It's precisely this. And there are a lot of subtle aspects about this. What's the right time scale to choose? If you noticed all the equations here, the time scale over which we calculate jump moments, the first jump moment and the second jump moment are not same. How do you find all of this? This is in a paper which I hope I have put the citation somewhere in the end. Any other questions?
No, there are no other questions in the chat. Ah,
Okay.
sorry. Yes.
One more.
Is,
Sure.
There is from Bert, who is asking, is the system size expansion a mean field model with stochastic term?
Absolutely right. So the mean field model, mean field part, deterministic part is identical to mean field approximation. The stochastic part comes from the fact that the number of individuals in a fish school are finite. And that comes, system size expansion tells you how to construct the functional form for the stochastic term, also accounting for the fact that there are only finite number of individuals. So if n was infinitely large, that term becomes 0. So I'll just show that again. Oops. This one. So you can see that the stochastic part has 1 over root n term. So if n was infinitely large, stochasticity becomes 0.
Great. Okay. I think you can move on.
There's another question. I can take that up later again. I think it's related to the previous ones. But I think this is a general question. This is a great question by Prajwal again. Maybe at the end I can take this. Is that okay, Prajwal?
Yes, I think we can move on and answer.
Okay. Okay. Sorry, where was I? I was in this slide. So now I have just showed you the theory and in principle how a method can actually help us extract these jump moments from data. Okay. Now I had already shown you experiments, right? So I had already shown that we were able to compute these jump, order parameter polarization as a function of time. Okay. For different group sizes. Can you apply the method of jump moments and construct the stochastic differential equation for these data? That's precisely what we did. And so this is the, okay, I have already shown you the result now. What we have done, we did exactly that. We did this for all the 3 group sizes simultaneously, and then we chose the best fit model. And this is the equation we found. And as you can see, the deterministic equation we found was actually linear. The stochastic term we found has, again, this mod m square behavior, which was exactly same as that of the pairwise stochastic copying model. Okay, it almost seems magical. It's actually true. So what we therefore infer from our analysis is that because the deterministic term does not have cubic terms, and the stochastic term also matches with the qualitative features of the pairwise model. So our fish, which we analyzed, must be interacting with one another using the stochastic pairwise copying interaction rules. Okay, and moreover, not only that, we also can rule out the fact that ternary interactions are dominant mode of interactions. So, I mean, of course, I think I should have been careful in writing this second bullet point. We can never really rule out what fish are doing. What we can say is whether they are dominant or not. So clearly it appears that the ternary interactions are not dominant. If they were dominant, we would have found traces of cubic terms in the deterministic term. So for those who might be interested, I can, the sort of, if you look at the goodness of fit kind of measures, one of the measure is called R square, and the R square value for the this model is close to 0.9, which gives us a very strong sort of, reason to believe that this is a statistically good fit. Okay, so again, just to remind, pairwise interaction model had these 2 very simple rules. There was a stochastic turning rate, right? And then there was this pairwise copying rate, where one fish just looks at one other fish and turns towards it at a rate c. and this rate C and S become part of the stochastic differential equation in this very simple way. Okay, so to summarize this part of my talk, what I have shown you is that fish that we studied actually show simple pairwise interaction, pairwise stochastic interactions. This is quite contrast to classic models of local averaging. For example, the Vichek model. For example, the Vichek model that I mentioned, that many of you might be familiar with, assumes that each self-propelled particles looks at certain unit radius and looks at the direction of every individual in that radius and finds an average and moves in the average with a small error, right? That's the Vichek interaction rule. Likewise, the cousin model also does something similar. It also has rules for attraction and repulsion. Okay, now in our analysis, we were focused entirely on alignment only. So I can't talk about what's happening in the domain of attraction and repulsion. But if you focus entirely on the effective alignment rules, what we find is that it's contrast to the classical models of local averaging. So fish seem to be doing actually even simpler than what the Vichek model actually says. And we also, provided a method to construct models from the data. If you have a high resolution time series data, you can actually construct a stochastic differential equation model from those data, time series data. And these are the 3 references for those who are interested. They're also all easily available from my website, the PDFs or the archive links. So for somebody who's interested in understanding how do you go from these simple interaction rules to stochastic differential equation models, this chapter provides the review of the theory and some methods. It's a more of a pedagogical article as a part of book chapter. And then this paper, the second paper, tells you how do you go from this bottom-up question. If you had observational data, how do you go towards the theoretical? How do you construct an SDE? And the third paper actually applies both of them, and to the specific fish schools we studied. They're called Etroplus or Karimine locally. Okay, so that's the summary of the first part of my talk. Do I have more time to cover one other ongoing work on collective behavior?
Sure, absolutely. I mean, we have time. You have still 10 minutes, but we can go a little bit beyond.
Okay, now let me present another work where we look at how collective motion happens and sort of responds under the context of predation. You may remember the video I showed in the beginning where there were really fascinating escape patterns of fish schools, starling flocks, and I think krills responding to predation. Okay, so if you think of understanding collective motion in nature, they're not clearly in a fish tank of the type I showed, right? Secluded, free from any sort of threats and so on. So there are while many of the social interactions do shape collective movement in the nature, there are habitat features, environmental features, and importantly predation that shapes many of the interactions between animals that in turn causes collective movement, right? So much of the work in the field has sort of focused from the middle box to the right side arrow, and the focus on the left side hasn't been as much. And to understand this, one of my students is looking at this beautiful charismatic species called blackbuck. These blackbuck live in grasslands, and they are native to Indian subcontinent. The males are the ones with dark coat and these beautiful horns, and females are the orange bodied and typically slightly smaller in size. And many of the herds in this species are female groups with a few males here and there. Okay, and they move from place to place. Many groups merge together. So there is no single group that maintains identity as a function of time. So they keep merging, splitting, and moving around in the grasslands. Okay, so what we did was, of course, to observe a natural predation event, and then being able to record high resolution data is very, very difficult. So what we did was the following. We flew drones at a height of 30 to 60 meters over the herds, and we record them for about 15 minutes. Midway through the recording, one of the students or the experimentalist who is doing experiment will approach this herd. And these are really shy animals. And what these animals then do is observe this human coming towards them, and they escape or move away from this approaching human. And this approaching human is moving very slowly, cautiously, to not to make sure that it's as less invasive as possible. So this is a sort of a perturbation to the natural state of the groups of black buck, but it also acts as a mild threat. So this could be, we thought we could use this to understand how animals respond to perturbations and threats. So here's an example of a video. I hope it's playing. I hope the resolution is okay for you also. So this is the drone footage. As you saw a few seconds ago, somebody from the bottom part of the video moved towards the center, and there is now another part of the herd from the top right where they're all moving towards the rest of the herd, and now they have all picked up a bit of a speed and they're all moving in the same direction. Okay, all of this happened because there was somebody who one of the experimentalists moved towards this herd from the right side. So I have shown you about 30 seconds to 1 minute of this clip, although the entire video is, we have recorded about 7-8 minutes before this event happened, and also how does this event really settle down? How do herds settle down? Okay, so what we basically do is, with this data, we again use various image processing techniques, and we quantify the group dynamics, group cohesion, group polarization, group speed, and how do these groups respond to this simulated perturbation, simulated threat. For example, who initiated the response? Are there leaders and followers in this response? So what we basically do is we plot the two quantities here. I'm showing you two quantities. So in the herd, we first plot a mean near neighbor distance. If this mean near neighbor distance is small, that means the group is very cohesive. If it's large, that means the group is now spread out. So we plot time series of this, and then we also plot the group polarization, how aligned they are, with one another. What we do based on these time series is we can clearly identify that there are 4 events in this time series. One is the pre-perturbation event. There is a initiation of escape, and then that in turn causes coordinated movement, and then there is a settling of the behavior. So those are all drawn using this, red bars here. You need to use combination of near neighbor distance and polarization. You can see this, two very clear bouts of coordinated motion. You can also see a bout of increased distance between each other when they were responding. Okay, so we have done this for about 60 to 75 videos, and the results I'm going to show today is only for a smaller number of them. So what we want to understand from this, is how did the information about the perturbation or the threat, how did that spread across the entire group? And that we try to do by looking at how does the change in the speed of individuals happen from one individual to the other individual. And what we do to quantify this, what we do is we first identify who are the initiators, who are the first to respond, and we identify the initiators by basically looking at the speed of among all the individuals whose speed suddenly changed, and that individual is the initiator. And then the laggers, who are the last to respond. And from the initiator to laggers, we sort of construct a network of how the influence is propagating in the entire network of these individuals. Okay, and secondly, we also look at, are there some individuals who are more influential than the other individuals as individuals are moving in the entire group. And finally, there are also individuals who perform movement sometimes irrespective of what everybody else is doing. They are called isolated individuals. So here is an example of analysis and the results. So the approach was from the from the towards north, and this was the group. The numbers here show the order in which they responded. So as you can see, there's a lot of counterintuitive facts here. One is that who is the first individual to respond, second, third, fourth, they're not necessarily very obviously determined by the spatial locations of animals and where the threat is coming from. And then what we basically therefore do, this is somewhat schematically, we look at the pairwise cross correlation between individual velocities for all pairs in the entire group. And then for each pair, we identify who was ahead of the other in the velocity changes. So the basic idea is that, if you look at pairwise correlation in the directions, if one individual is always turning before the other individual turns, you can assume that the first individual is then the leader of that pair. So we do this for each pair in the entire group, and we sort of rank the strength of this relationship by the strength of correlation. So I'm going to show you two examples here. For example, if there are three individuals, A might be influencing C, C might be influencing B. In which case you say A is the leader, C is the second, the next follower, followed by B, right? On the other hand, it could be more complex. A might be influencing C and B. C is influencing only B, in which case A influences C, influences B, but A also influences B, right? So you can now construct a network and the strength and a network where the nodes are individuals and the arrows and the strength of arrow represents how strongly they are influencing each other. Okay, so here are the four networks that we find corresponding to four different stages of the experiment. This is the pre-perturbation. As you can see, the network really has no structure. Everybody is sort of independent. There are no clusters in the structure. And then there is an escape bout, right? And in escape bout, there are some that are initiating the movement and then they become correlated with the others. And those are all here in this greenish, this blob here. And then finally, when they become coordinated motion, you can see clusters. There are sort of small clusters that are moving in a coordinated ways. And then when there's when they all sort of come to some kind of a post-perturbation equilibrium, it again looks more like the pre-perturbation event. So we can actually construct these networks of how the information is flowing. And I'm going to just summarize what are all the results we have found, very preliminary results. These might change when we analyze more rigorously. One is that we basically find that the group as a whole speeds up first. So imagine you are an animal in the group. Should you first orient with your neighbor or should you first run? This might be one simple question you can ask. What we find from this analysis is that the groups usually first speed up. As a consequence, they seem to orient with one another. Okay, so and the third effect usually is that of being more cohesive. Because they're speeding up and orienting, then you find that the group is now more cohesive. Okay, and then secondly, we find that there are these leader-follower relationships that happens only in this escape event and not in the other three events. And finally, there is a lot of variation, between individual responses. For example, the ones who initiate the response are not necessarily same as the influential individuals. The one who has most influence on the movement of others need not be the ones first to respond. So that's another interesting results. So these are all some preliminary results of how we can understand collective motion and pattern formation in the natural conditions. So that's sort of the end of my talk. So the work I presented today, the first work, was done by PhD student Jitesh Chawar, who is now a postdoc at Max Planck Institute in Constance. And the second part of the work on black buck is done by Akanksha Rathore. And of course, there are many collaborators involved, like in any other project. So that's the sort of end of my talk today. Thank you, everyone. And I can now take more questions.
Great. Thanks a lot, Vish, for the very nice talks. So there was the question by Prajwal.
Yeah.
That perhaps we can address now. I'll read it so people in YouTube can know it. So Prajwal is asking, what about cases where you don't know what the average rules are? So the previous question was, sorry, about distinguishing between interaction rules coming from top-down approach and sort of the connection between theory of and data. And this question is, but what about cases where you don't know what the average rules are? I guess I'm thinking more about the Vichek model and whether the jump moment still remains cubic or changes to something else. When looking at the data, we wouldn't know if the first jump moment will be cubic or linear or something else.
Okay, so so Vichek model also has a cubic term. So if you look at the mean field Vichek model, it's exactly same as a ternary interaction model, which is why I was interchangeably using ternary and Vichek, ternary and average. Ternary also has an average interaction model. So, but you're right that, okay, more generally, if there were some complex interaction rules for which you cannot analytically determine such a closed form solution for the jump moments, what do you do? I don't know. But that problem is a very universal problem, right? So see, there is no way we will ever observe what any individual or cells or animals are doing, right? We're only going to look at some coarse grain property, and we measure that, and then measure something about the coarse grain property, right? And then we see which of the models best explains this coarse grain property. And in this case, it happens to be the stochastic differential equation itself. So yeah, so I hope I don't know if my answer is very clear either. You're right that when we're looking at data, we wouldn't know, but we can construct from the data, as I mentioned, right? I hope that answer was clear. Now, how do you construct equation from data? Was that clear, by the way? Because that's very central to what I said. Because if you cannot do that, there is no way you will be able to distinguish the models.
Yes, there is no sign in the chat from Prajwal, but I guess it's a sign that it's clear. So we have time for another question. If anyone, okay. Okay, there is another point made by Prajwal. I think you can answer these and then we can move forward. So the previous answer is clear. Thanks. I have a set of questions. Smiling face. One, in the models, you show that most other models, the speed is considered constant. But in the black buck case, that's not true. How will this, changing this hypothesis change the model and the dynamics? So then there is another question. In the same group, if you repeat the perturbation multiple times, do the leader, individual, and the hierarchy stay the same? And in line with the previous question, if repeating multiple times, how would one control for all sorts of different effects like memory or learning? So I guess the question can be summarized in whether it's true that the speeding constant and the hierarchy is constant.
Yeah. So you're right that in the simple model, the speed is constant. So there are some groups which have developed variable speed models. We also have developed one that is sort of driven by the data we have. And it does show some surprising results. So it depends on the question you are trying to address, whether the variable speed is important. For example, even the fish have variable speed. Right? It's not that they are always moving at constant speed, right? But if the question is the polarization dynamics, maybe it's not very important. Okay. But if there's some other question in the context of prediction, speed does become important. So therefore, in such cases, one has to certainly change the model. I fully agree with you. I mean, that's also true for black buck in the sense that the very thing we are measuring really is changes in the speed. So coming to second and third questions, can you apply this perturbation multiple times to the same group? You're absolutely right that the learning will play a huge role when you do this. So therefore, we don't apply perturbation to the same group over time. So we always try to ensure that we have found a different group and we don't disturb the same group repeatedly. So memory, a lot of other factors play a role if you repeat the, the same experiment or the same group.
Great. So I think we can stop here. I don't see any other question. So I'd like to thank you, Vishu, again for the 3 great lectures. And thanks all the participants for their participation. So the next session is going to start at 2:30 Italian time. So we have more than 1 hour to take a break from the screen. And then we'll be back with 2 panels and the last 2 lectures by Justin Nickel and Alvaro Sanchez. So thanks everyone again. Thank you, Vishu. See you in 80 minutes.
Thank you, everyone, and thanks for the invitation, Jacob.
Thank you.
Bye.
Just a technical information, the Zoom meeting will stay open, I guess. So if you want, you can stay connected. But you can also disconnect and reconnect.
Simon, because of another meeting.
Yes.
Ciao Matteo.
Ciao Andrea.
Tutto bene?
Yes, yes.
Welcome everybody to this session. So we are waiting for the panelists to join us. So we'll start as soon as all the panelists are here. If you just connected, we are just waiting for all the speakers to and the panelists to join. And as soon as they are all in the meeting room, we'll be able to start with this second roundtable.
Okay, sorry, I'm back. So I've been contacted by Simon. Is having some technical problem, but he's going to join us as soon as possible. In the meantime, he asked me to introduce, to welcome everybody and to start the session. So I would like very much to thank all the participants of the roundtable, particularly Partha Dasgupta, whom I don't see since a long time. He's a long-time friend of ICTP. We had a very fruitful program on ecological economics. And then, well, Andrea Rinaldo, Alessandro Tavoni is
that's fine.
Kanchan also joining for this.
Thanks everybody for
yes.
So we've scheduled.
So Jacopo, you want to say something for
this seemed like a good
to
opportunity.
start?
I think we can proceed with the panel. I'll say some general announcement about the school later.
Your circumstances are,
so
to know you,
the
understand your mission
so the idea is to first introduce the panelists. So maybe we can have a round of introduction by each of you. Maybe we can start with Partha. You want to start?
Well, thank you very much. It's lovely to be here. Look, there is a background noise coming. Somebody's speaking. Is there a translator at work or what?
Yes, maybe it's better. Maybe it was me. Ah, yes. Sorry. Is it better?
Yes, well, yeah, that's fine. Yes, thank you. Thank you very much. I'm Partha Dasgupta. I'm speaking from Cambridge, and as Matteo said, we worked together many years ago, 2 decades ago, at ICTP. He was hosting a program that Simon and I and Carl-Johan Mailer ran for 4 or 5 years, I think, at the time of early 2000 on ecological economics. And I'm delighted to be here. Where is Simon?
Simon had some technical problems, but I think he's going to join us very soon.
Okay. Anyway, thank you.
Alessandro, you want to go ahead?
Sure. Thank you, Matteo. My name is Alessandro Tavoni. I'm connecting from Bologna, where I'm based in Italy, the University of Bologna. I'm an economist as well, an environmental economist. I've worked with Simon and colleagues, so I have a bit of understanding of complex adaptive systems and ecology as well, but my main area would be environmental economics and behavioral economics. So I try to address environmental dilemmas using these tools, and we can talk a bit more about it as we go.
Okay, in the meantime, Simon joined us. So Simon, we started introducing the panelists with Partha and Alessandro. Then I'll leave you to go ahead.
Good, good. Now maybe we can turn to Karina and Kanchan. Karina, you're muted.
Okay, can you hear me now?
Yes.
Yeah, good. I'm Karina Nyborg. I'm speaking from Oslo, Norway. I'm like Alessandro, I'm an environmental economist who also does a lot of behavioral economics. I've been working a bit with Simon and other ecologists in connection with the Bayer Institute in Sweden. And I'm looking forward to this.
Very good. Kanchan, I assume you're on somewhere. Don't hear her. Is Kanchan Chopra on? Let's come back to her and go to Andrea Rinaldo.
Yes, thank you, Simon. Good to see you. Andrea Rinaldo. I'm a professor at the École Polytechnique Fédérale in Lausanne, but right now I'm in Italy, my home place. I'm in Padua now, and you see a piece of my beloved hometown, Venice, in the background during the lockdown. And my work is on water controls on biota and I'm delighted to be here.
Very good. Mercedes, are you on?
I think Mercedes has not joined yet.
Okay, Kanchan, I'm glad to see you there. Why don't you, we're just introducing ourselves. Could you just say a few words about who you are and what you do? I don't know if Kanchan can hear me yet. Kanchan, can you hear me? I'm sure we'll get these problems sorted out. Kanchan, can you hear me now?
Hello.
Hello.
Hi.
Hello.
Hi. Hello. Hello, Simon Kanchan here.
Yes. Can you hear me? Kanchan, can you hear yet?
Yes, I can hear you. I can hear you.
Good. We're all introducing ourselves, and just could you just say a few words about where you are and what you do? Although I think most of us know, but go ahead.
My name is Kanchan Chopra. I work in Delhi at the Institute of Economic Growth, or I'm associated with that now. Was on the faculty and was director for a long time. My area of work, of course, environment and development. And in the course of my work, I have been privileged to look at different aspects of the relationship between environment and development. I have been at ICTP Trieste for a number of summer schools and winter schools. And I think that's all that I would like to say at the moment. Thank
Very
you.
good. Thank you. Thank you. Thanks all for joining us. Matteo, I assume you introduced yourself.
Well, no, but unfortunately, as I mentioned to you, I have to leave for another meeting. So I'm very sorry about this, and I wish you a very interesting panel discussion.
Very good. Thank you all very much. So as most of you know, except for the panelists, in the lecture I gave Monday, I made the argument that building stronger interfaces with economics and the other social sciences was one of the great challenges facing theoretical ecology and any ecology that has to do with the environment in the next century. There are a number of cross-cutting issues I'm hoping we will touch on today. How do you value nature? How do we discount the future? How do we deal with equity within populations, across time? How do we get cooperation in dealing with problems of the commons and public goods? What's the role of pro-sociality and social norms? How do we incorporate the dynamics of human behavior during crises like pandemics, which was the subject really of yesterday's roundtable? But I'd like to begin by trying to filter all of this through one of the fundamental issues that faces us, namely the decline of biological diversity. And the most influential thing that's being done on this topic in a long time is the so-called Dasgupta report that Partha is leading for the British government. So I'd like to begin by asking Partha, my longtime friend and colleague, to tell us what that project is and what the issues are in biological diversity that ought to interest theoretical ecologists, applied ecologists, economists, and all those working at the interface. Partha.
Thank you very much. Can you hear me? Is it? Am I?
Yes, very clear.
So the UK Treasury invited me last summer, that is 2019 spring actually, to produce it's a independent global review of the economics of biodiversity. So there has been, one of the nice aspects of this has been that there's been no influence from government circles as to how I conduct the review, what is said in it. It's entirely a personal view. Of course, I've been helped by a team at the Treasury, goes that saying, who are an extremely talented group of young people, self-selected because they, I think, applied to be on the review team. So it's been a great experience. Now the review is completed now, and it's gone to the senior ministers of the Treasury and DEFRA, the Environment Ministry, and it will be formally launched on February the 2nd, I think, 2021, in a month and a half's time, at the Royal Society, where Venki Raman, the president of the Royal Society, will be the host, if you like, with members of the cabinet, I guess. So what is it about? Well, first of all, although the review is on biodiversity, one should remember biodiversity is a character, a property of ecosystems, or whatever system, natural system you're studying with biological trappings in it. It's a property, it's not an asset, it's a characteristic of an asset. So the intuition that ecologists such as Simon and others have had all these decades, that we ought to be looking at the biosphere at the top to the smallest allowable notion of an ecosystem as a component, was exactly right. There are many ways of defining biodiversity, but once you start going in that route, we're in trouble, because it'll become like sociology, define your term kind of thing. So we're looking at ecosystem services. This has been much work done on that, and we start from there. Now, the position is the idea is to regard nature as an asset. I'm using the word nature really as a simple way of conveying the idea that it could be the large macro study, or it could be a small ecosystem, maybe a threshing ground or a forest plot, depending on the context. So we're looking at assets on a par, more fundamental, of course, but in the same vein as produced and human capital. We're looking at natural capital here. And so we convert the entire problem as an asset management problem, whether you're a first private investor, or whether you're a social reformer, or whether you're head of some para-national institution. And so it's built on that. The value itself need not necessarily be use value. It could be intrinsic value. Societies, all societies one can think of, has to regard some assets, some natural capital, as sacred even. So that shouldn't deter us from thinking of it as an asset. Once you recognize that the worth has many dimensions to it, once you recognize that, then you're really talking about the economics of the biosphere. It's as large a subject as that. What we show, the theorem that really drives the whole, unifies the treatment of the economics of biosphere, is the equivalence between what we call inclusive wealth and intergenerational welfare. These two objects have different sources. Welfare is a normative notion altogether. And I'm talking about intergenerational one, adding up maybe the generational well-being or some nonlinear function, doesn't matter. That's not the important thing. The important thing is that you're including future people in it. And whereas wealth is a value of stocks, assets. And that seems more like the means to the ends. The connection between the two, the equivalence, lies in the prices, the values that you attribute to the assets. So Simon was exactly right in thinking that valuation becomes an important part of the problem that ties, if you like, the means, which are the assets, and the ends, which are reflected in well-being, human well-being, or indeed the well-being of nature. That's not ruled out at all. If there is intrinsic value, If it's sacred, they'll have a value independent of what it does for us. So the approach we take is much broader than is usually given in environmental and resource economics, but that's not a weakness of environmental and resource economics. It's very useful to have use value, human use value as a start. And the reason is that one thing we point out over and over again in the review is that using biased estimates can be very informative. If use value alone tells us that we must conserve nature, then of course, if you add non-use value, intrinsic value, there'll be an additional reason for conserving nature. So biased ones are not to be dispensed with. Okay, when I say the equivalence of these two ideas of inclusive wealth, which goes in the direction of national accounting, if you like, suggesting that national accounts now ought to move away from flows to balance sheets, if you like, of firms. And that the overall thing is an asset management problem tells us that the equivalence result is telling us that the aim of the game is value maximization. That is, whoever it is who is managing a portfolio, whether it's the personal investor or whether it's a social reformer, doesn't matter, the whole range can be accommodated. They will be using different prices, obviously, market prices for the investor, private investor, social prices or accounting prices or values, social values. If you're a reformer, citizen, thinking as a citizen, you're interested in maximizing the value of your portfolio. And that's the same thing as saying maximizing wealth. So wealth maximization turns out to be essentially a portfolio management instruction. So that unifies both macro economics of growth and development to very, very micromanagement of assets. So that's, broadly speaking, what we are concerned with. Now, I want to end with just as a preliminary remark, a final observation. What makes this area particularly interesting intellectually and deep conceptually is that nature has three properties which are not usually shared by other forms of human capital. First is, of course, the well-known property of mobility. Nature is always on the move. And mobility leads to these externalities, which we all understand very well. We economists understand very well, and with ecologists have done a lot of work on it. I want to leave that aside for the moment. There are two other features which the review emphasizes. One is that nature is often, very often, silent. Natural processes are silent. What's happening under your feet in the soils are not audible. You need extremely precise instruments to go to that. On a daily basis, you can't. And the other is that it's invisible. So invisibility and silence of nature makes monitoring of human activity extremely hard, of what we are doing to nature. In fact, it makes it impossible. So the review ends by suggesting I worked through serious issues of institution, what can be observed, what can be verified, how far the law can handle matters, how far social norms can handle matters. Those are discussed in detail, but invisibility and silence means that there will be a residual aspects of our activities which will not be monitorable, no matter how good your institutions are. So in some sense, the review points to the limits of institutions as a way of bringing our private incentives aligned with our citizens' goals, our goals as citizens, which means that there will be a residual amount that will not be observable. And the only way we can manage to live at peace with nature, which is another way of saying at peace with ourselves, since we are embedded in nature, we're not external to nature, is restraint. In other words, we have to be, at the end of the day, both judge and jury of our own actions, never mind other people's actions. So the review ends with a plea for a reform of education. It suggests we appeal to the idea that nature studies should begin right at the beginning as primary school, and not to be dropped as we go through our educational process. In effect, it's appealing to all of us to become in part naturalists. I'll stop here then, and we can take the discussion.
Thank you very much, Partha. Before I turn to the other panelists, could you elaborate a little bit on how this framework deals with the problem of discounting the future? This has been a subject of debate, even ignoring hyperbolic discounting. How do we decide what's the right discount rate to use?
Okay, that's very good. We are really looking at intertemporal prices, I suppose. That's about what we mean by discounting. It's an asset management. We are still at the level of asset management. But typical asset management problem, portfolio selection, is concerned with managing the assets at a moment in time. We've got portfolios to choose between, and we look at today's investment, tomorrow's realization, and then we can reorganize our portfolio. The question that Simon is asking relates to a further problem in portfolio management, that is portfolio management over time. Typically, when you think of a portfolio manager, he's got a certain amount of assets, wealth to invest, and then he's choosing between alternative portfolios. But the intertemporal problem lies in asking a prior question: how much should he consume and how much should he actually invest? So that too is a portfolio problem. And what we regard as first order conditions, Euler conditions in optimization exercise is a portfolio, is an arbitrage condition. Okay, so that's the background. I take the position, as we all should, that accounting prices or shadow prices or social values are far more subjective and far more amorphous than market prices, which are much more real. For these accounting prices, especially about the future, so much of it is based on the decision makers' understanding of what is feasible and what is desirable. These things cannot be aggregated across all people. People will differ, and in a democratic society, we can argue about them, we can discuss it, but the idea that there's a hard stuff out there which we pluck out of air, that's out, that's not on. So social discount rates, which are intertemporal prices, price of future goods compared to today's goods of the same good, let's say consumption or investment, doesn't matter what your unit of account is going to be inherently a subjective element. And if I say it's 4% and Simon says it's 2%, there will be a reason for us to have a discussion as to how we have come to our conclusions, or not, if what our conclusions are tentative suggestions. And the discussion may illuminate, we may in fact converge. That's the stuff of debate on matters which are not hard. For the private investor, the discount rate may be the market price. Okay, well then, he's small, he can't affect the prices, so he takes the prices as given. But we're not looking at the problem from the point of view of citizen, because Simon is asking about social discount rate, not private discount rate. And there we have a problem, in the sense of problem at one level, and another level it's not a problem. A good, diverse, democratic society will have disagreements maybe, but we resolve those disagreements through the political process. We don't shoot each other or persuade each other that we are all in agreement when we may not be. So what does it really amount to? It amounts to the idea that we want to internalize these externalities over time. And I'll finish here by just reminding us of the short circuit that economists have so far made, which I think has been extremely dangerous, damaging, because it's faulty science, in pretending, here's the idea. The idea is, well, look, we all care about our children, and if we're intelligent, we realize our children will care about their children, and therefore, by recursion, if we're intelligent, we'll be caring about our own grandchildren, great-grandchildren, and so forth. Okay? So why not leave it to the market? Why not leave it to our own judgments? And that's been used very decisively in American welfare economics, the idea of a representative household as reflecting an entire economy. Now, the problem with that argument, and it's really astonishing that it could in fact survive so long, even in the case in a world in which you're studying externalities like global climate change, is that I may care about my great-grandchildren with great unerring accuracy, and Simon might do the same with his grandchildren. But there is no reason to believe that he will care equally about my grandchildren or me about his grandchildren. That's the whole stuff about externalities. So that route is not on. We really cannot use market rates of return for social discounting for this externality reason, which brings me back to the point that really, at the end of the day, as citizens, we then say to ourselves, I say to myself, I must take into account Simon's grandchildren. And Simon, as a citizen, says, yes, Partha also will have grandchildren, and he should enter my calculation when I think about social discount rates. But there's no obvious reason how we should necessarily come to the same rate, which is where I began by saying they're going to be ultimately somewhat subjective. They won't be arbitrary. They won't be wild. If somebody says it's 100%, oh boy, we will ask him, really pin him down and say, go and explain where you get 100%, and all the trade-offs that are involved. Okay.
Thank you very much. Karina, I'd like to turn to you next.
Right. Sorry, I lost my headphones. Now I can hear you.
Yeah, I'd like to turn to you now.
Yeah.
For comments on what Partha has been telling us.
Right. So I think that what Partha said earlier on the importance of non-observability is a very, very important point. And he said a lot of what I would have said myself, and he said it in a much better way than I could have done. But I think, economists have been working with environmental and ecological economics for many, many years. And for a lot of the problems that we face, economists have good solutions. But one of the main problems that we don't really know how to solve in a centralized way, at least, is the problem that often we cannot see what people are doing. And if we want to limit the depletion of natural resources, then we need, and we want to do it in a centralized way, we need to somehow be able to see what's going on. And if nature doesn't shout out when she's being exploited too much, it's hard to do that. So I think one of the things that economists don't really have very good centralized policies to solve is a lot of the problems linked to this non-observability. And I think we cannot really get around the idea that Partha is advocating, that partly we do. I mean, even if politicians listen to economists in their suggestions of how to solve pollution problems, for example, we cannot really solve environmental problems satisfactorily if people don't feel a moral obligation, social obligation, but also a sort of internal moral obligation to take good care of nature. And I agree with Partha that a very important tool to try to make that happen is actually to start education about nature very early on. But also it is possible that policymakers can have a role in shaping social and moral norms. Education is one thing, but I have also been concerned about in my own research that a lot of what we do in society is shaped by, at least partly by, our need to coordinate our actions. So, so often it is hard to change your own behavior because you need to coordinate with what other people are doing. And policymakers can often help change the ways we coordinate. Social norms can be one example of such coordination. We often share values, share habits, and sometimes you can sometimes there will be multiple equilibria, and we are in a bad equilibria when it comes to protection of nature. And sometimes it is possible for policymakers to contribute to flip the economy to a better equilibrium. For example, think of our diets. If you live in a society of meat eaters, then it is very inconvenient to be a vegetarian, because every time you visit somebody or you're sharing a meal with somebody, you feel you're making trouble for the others, and the others feel you're making trouble for them. But if you live in a society of vegetarians, it is the meat eater who is causing problems and getting problems, and thus may not be invited, for example. So, and to get from one situation to the other, public authorities may have a role in, for example, temporarily subsidize vegetables, tax meats, things like that, and also promoting policies that can help change habits.
Okay, thank you very much. By the way, I have put a link in the chat to the current UK government site for the Dasgupta review. That part of it is public. Partha, it's not yet complete, is that right?
Oh yes, it's complete. It's been sent. As I was saying,
okay.
Our obligation was first to send it to the senior ministers, and copies have been sent also to the advisory panel that the Treasury had created for us for my review. But it will not be made public until it's been handed over officially, some protocol with the finance minister, and that'll be on 2nd of February. The moment it's done, it'll be on the website. All three. I should say there'll be 3 documents. One is the review itself, which is a technical piece of work. It's about 600 pages long now, because, as I say, it's the economics of biosphere, and a good deal of economics had actually to be redone to make it in line with what I was sketching for you, including the points that Karin made. The points I was making are somewhat different from Karin's, because even those, they're observable. I can watch the person eating meat, or the meat eaters. I wasn't thinking of that kind of coordination problem. I was thinking about things which literally cannot be monitored by anybody. What I do is entirely private. That's the bit which I don't think will ever be resolved, except through self-restraint. So that's the first, and it has. I've tried to keep the text in English, even in the or text, if you like, but it has lots and lots of boxes, annexes, start chapters for the mathematical parts of it, because everything is, as far as possible, proven as in general form. But on the other hand, I wanted to reach the general public, the citizen, concerned citizen, my ideal reader. And so I prepared a 100-page abridged version of it, and that will also be on the website, and it'll be published alongside with the main text. And then there is a very short 4 or 5 page, if you like, headline messages that my team produced, because that's necessary for the decision makers in government, since the report is ultimately for the UK Treasury and the government in general, and of course it'll be used by the Treasury and the government for the COP15, which will be held next year in China. That's the UN biodiversity, the committee involved with biodiversity, in parallel with Climate Change 26, which will be held in the UK next year again. So we are making some progress in this area, but this is the foundations for the biodiversity one. So it'll be February the 2nd. I think it'll be on the website, all 3 documents.
Thank you very much.
Simon, can I chime in a bit on that?
Yeah, sure.
Yeah, so I perfectly agree with Partha that he was speaking about something else than the coordination problems that I spoke about. So that's actually why I spoke about moral norms and social norms as different, because what I think of as social norms is requires some kind of observability by your peers. It's not something. What you do is not necessarily observable by the central authorities, but it might be observable by your peers. And in that case, you can have social coordination or social norms, if you're lucky, help solve part of the problem. But I perfectly agree that a lot of these problems cannot be solved that way, simply because even your peers will not see what you're doing. And then we need internalized moral norms.
Right. Very good. I should say that I mentioned at the beginning, the social norms issue is a huge one. Karina and I were part of a paper a few years back. Alessandro, I can't remember if you were part of that or not. But we talked about it yesterday in terms of mask wearing, and it's going to come up again in terms of vaccine hesitancy. It's a cross-cutting issue. Kanchan, I'd like to turn to you next, for you to give us thoughts on any aspects of this or other issues that you'd like.
Thank you. I'll do that. I'm fine with that. Well, there are several challenges at the interface of ecology and the social sciences, in particular economics. You wanted me to talk a little about natural capital accounting. I'll come to that. In the space of the last 2 decades, we've had a whole lot of general frameworks coming up. General frameworks or grand designs that provide space for linking the social sciences, in particular economics, with with ecology. And of course, in the beginning, we thought that ecology and economics should be most easily thought of as having a common mathematical language and easy to link to each other through that language. But they have to come out of their comfort zones. And context dependence makes it difficult.
Oops. I think we'll have to come back to Kanchan. She seems to have frozen now. Kanchan, you've frozen temporarily.
Some of these. Oh my! I'll come back. So now.
Yes, we can hear you now. But you keep freezing. Maybe closing your video and just coming in just by sound would work. Now you're muted. We have your video, but
yeah, I've unmuted myself. I'm sorry for that. I don't know what happened. And actually, some developments, including attempts to capture climate tipping points in damage functions in econometrics and so on. And we have other movements like the planetary boundaries approach. But more than that, when it comes to inclusive wealth and systems of national income, we want to link the systems of national income to what is we call natural natural capital accounting. Now, how do we do that? That is the context dependence, which I'd like to talk about a little, which is really in the sense of suppose I want to find out a part of the different kinds of capital is what inclusive 6 or 7 years ago, which was chaired by Partha Dasgupta, and there we had referred to the aspects there. But you have to get an estimate of the value of ecosystem services. Now, in order to value these ecosystem services, we need to know basics like, for instance, production functions of life supporting systems. Now, for economists, production functions are important. But when it comes to life supporting systems, it's very difficult because spatial and temporal boundaries of specific ecosystems need to be identified. Spatial and temporal boundaries of specific ecosystems need to be identified in order to integrate them, in order to get the value of that ecosystem service. As a grand design, it's fine. We will say we'll value the ecosystem services. But in order to do that and to integrate with systems of national income accounting, you need to know the boundaries of that ecosystem. And that is where it is a tough job. And that's one of
I think we're frozen again. We'll come back to Kanchan.
Actually integrate side, looking at short-term impacts of development. Fine.
Kanchan, can you hear me? I think we're going to need to move on. Alessandro, I'd like you to take over now and give us your views, and I'll see what we can do about Kanchan.
Okay, let me jump in, although it's going to be a tough role to follow such eloquent speakers. But I'd like to touch on some of the topics that emerged with Partha's discussion and Karin's points about social norms. As I mentioned, I'm interested also in the behavioral aspects behind cooperation. And as we discussed before, oftentimes we simply do not observe, either because nature does not scream and call our attention, or simply because our own actions are not observable themselves. And in the situation when the social norm is low, meaning that that behavior is a niche, the pro-social behavior, the environmentally friendly behavior, is still so small in the population that we're talking about a niche behavior. How do we scale it up? How do we ramp it up? I think essentially what we saw before is that there is a role for government to intervene, and one channel could be, and I agree with what has been said by Partha and Karin, that education, instilling such inclination, proclivity to cooperate and to appreciate the environment is key. I think in some of these situations, we can also have help from interventions that may not necessarily come from a government. And if you think of the economics profession, a lot of the interest has traditionally been on self-interested motives. We discussed briefly before about wealth maximization and these sort of drivers. However, there are other motives. We know that from ample evidence from social psychology, and also behavioral economics. And some of these altruistic motives can be leveraged also when it comes to cooperation in climate change dilemmas or global public goods. So to be concrete, we were discussing the situation when a behavior is not observable. That is the most problematic, right? If I switch off my lights at home, or if I have a renewable energy contract that arguably is a little bit more costly, no one knows about it unless I go about and talk to my peers and friends and family and try to convince them. The economic interpretation of pro-social behaviors tend to focus on what is known as image concerns or social signaling. That again is not an altruistic motive, but it is a self-interested one. I do something pro-social because I hope that by taking that action, third party observers, my peers, those that can actually see this behavior will have a higher esteem or will return my behavior with some kind of reciprocity. Now, what if the behavior is intrinsically hard to observe? Again, there's a role for government or firms, businesses to try and modify this situation. So again, we are in a coordination, in a bad equilibrium, because people struggle to witness, to observe potentially inspiring pro-social behaviors by others. And simple nudges and interventions, again, at the firm level or mandated by the government, can reduce the cost of me individually going around shouting that I'm great because I have a renewable energy contract, which also can be seen as sort of socially costly, right? It could be perceived as bragging. So an intervention may aim to make it public, to make the decision public to others, so that imitation can kick in. And again, I have to flag that in this domain, and here we go full circle and go back to education. I think that the economics profession can do more in terms of training future policymakers, future academics. I see it in my own university. I'm at the economics department, and the students tend to not be introduced interdisciplinary concepts, or even just basic knowledge about climate change or even environmental economics until later in their curriculum. And again, the focus of the standard curriculum, if you take sort of basic economic courses, tend to be on these self-interested rational, but psychological evidence suggests that it is otherwise, and we have an innate proclivity for cooperation. And so I think also by teaching that there may be more and stressing these sort of leadership and altruistic motives also in the standard curriculum could help out in bridging the gap that Partha so eloquently described in the introductory talk.
Thank you very much. Andrea, I'm glad that we can turn to get a perspective from outside economics, but from someone who recognizes how important these issues are. So be interested in your thoughts on what we've heard so far this morning.
Thank you so much, Simon. Yes, indeed, I'm very interested and very passionate about those subjects. As you hinted, I see things from a narrow perspective, that of the perspective of water controls on living communities. And of course, not all ecosystems are water controlled. Many are light limited, many are nutrient limited. There are there are several things, and yet but many are, and in fact, I'm advocating for the social and economic importance of many of those ecosystems, and for two different reasons, which I'll be discussing in a second. I am obsessed, in fact, and even from our early work together, Simon and with Ignacio Rodriguez Iturbe, in fact, from the inherent weakness of many of our ecological predictions, and that's, in fact, it's a permanent liability on our capability to put a price tag, in fact, on ecosystem services. And so, well, I'm very touched when you say that about those characters that Partha was eloquently pointing out, that is the silent, invisible characters that we may have beneath our feet, something that cannot be monitored, but perhaps can be profitably guessed. And here I'm not quoting a scientist, but I'm quoting a poet, Auden. If I'm quoting by heart, I think I'm correct when he said that knowledge may have its purposes, but guessing is always more fun than knowing. And why am I pitching for the water controlled ecosystem? Because there's something about the substrate for ecological interactions. There's something about what we know about what economists call externalities that can be useful to predict shifting patterns on rainfall, mean a hell of a lot for rain-fed agriculture, which is a major driver of social well-being on a global scale, in fact. And we know a lot about patterns of rainfall and how they affect ecosystems and ecosystem services. And the accounting for the depreciation of natural capital, in fact, has some sort of inherent robustness when you're talking about water controls and on those living communities. So well, typical questions that we'd like to point together, it is, for instance, water management schemes and large-scale water management schemes mean a hell of a lot. Will future large-scale water resources management plans include biodiversity protections worldwide? Of course, to do that, we need to be sure that our guessing is complementing knowing in a good and meaningful sense. Could, for instance, the structure, something that we have studied together, Simon, for a long time, and with Ignacio too, like the structure of the river network be a template on how deadly waterborne disease can spread? And something else which is also important, can we judge whether we can sort out how the impact of improved agriculture, for instance, with its well-documented inroads into the spreading of the incidence of debilitating disease, for instance, be accounted for properly? Can we, and that's central in the reflection that it's a quantitative example of what Partha was saying when accounts that like gross domestic products of ad hoc indicators of economic well-being do not represent the depreciation for natural capital. So in the GDP of Burkina Faso, where we did a hell of a lot of field work, for instance, 15,000 small dams have been built and irrigation structures have made a visible impact on the GDP of this peculiar sub-Saharan country. And yet the dramatically increased incidence of debilitating disease is not accounted for in any of those indicators. So in fact, biological invasions is another example. There is an inherent predictability about the biological invasion of alien species and whose economics can be done based on the fact that we can actually guess relatively well the possible scenarios in terms of what happens if and what happens if. And that happens to disease spread, actually. So what I'm saying is that seen from outside, and seen from a narrow perspective, the one I can't command, but I see that those issues would be central with respect to what we call eco-hydrological research, that is research of living communities controlled by water. Not again, once for all, it has to be clear, an entire view to the ecosystem perspective, but certainly a narrow but significant one from a social and economic viewpoint.
Thank you very much, Andrea. So we're just about out of time. I want to thank all the panelists. We've just touched on a lot of the great challenges that I think face us Partha talked about biodiversity, the issue of discounting, which we just barely scratched the surface on. We didn't talk much about equity, especially intra-generational equity, which is a crucial problem. We heard about natural capital accounting from Kanchan, and about issues of cooperation and social norms, both from Karina and from Alessandro. This is a rich interface in which not only are there a range of problems that are at the boundary between ecology and economics, but a recognition that there's, as Kenneth Arrow pointed out, it's not an accident that economics and ecology both start with eco, which means household. In the case of economics, household management. The problems that we face are similar. Public goods problems are prevalent throughout the natural world, and in things as simple as the extracellular polymers that biofilms produce, the public goods problems that tumors undercut, and bacterial biofilms that form all the way up to cooperation in bird flocks, fish schools, and up to humans. So they're not only interfaces, but great parallels, which is why, not surprisingly, many of the similar techniques, especially game theory, have permeated both disciplines. So take our word for it. This is an exciting area. There's much to be done, both at the interface and by drawing parallels. We could go on for 3 hours easily on this, but I want to thank again the panelists very much, and now close the session. Thank you.
Ciao. Ciao, Simon. Ciao, Partha. Ciao.
Thank you very much. It's a pleasure.
My pleasure too.
Thank you.
Thanks.
Thank you. Sorry for the technical
It's not your...
glitch, but anyway, that's fine. I'm sorry for the interruption.
Not.
I could anyway. Thank you. Thank you. That's
Thank you.
it.
Great. So thank you very much, Simon, and to all the panelists for this very interesting session. So we are now taking a 15-minute break, and we are going to be split in the breakout rooms. So use these 15 minutes to take a break from the screen, to chat with others, etc. And then we're going to be back at 3:45 Italian time with the last lecture by Alvaro Sanchez. Thank you very much.
Huh?
Just a reminder, we'll be back in a few minutes. Okay, I think that everybody should be back in the main meeting room because I don't see the breakout rooms open. So before we start with the next lecture, well, I want to make a few announcements. I mean, as you know, we have 3 slots left, a lecture by Alvaro Sanchez, a lecture by Justin Nickel, and final roundtable. Given the sort of interest in having more space for discussion that emerged sort of at the side of many talks, we decided to, well, after a break from Zoom and screens, to have a sort of follow-up workshop, which will take place in January. That will be much more relaxed in terms of schedule. It will be 3 days, 3 hours a day, short talks by junior speakers, and a lot of time for discussions. The title of these workshops is The Limits of Diversity Assembly. And I think you all will receive an email with the details to apply to this workshop as well if you are interested. So I just announced that there will be something to follow this long marathon of this school. I will also pass in the chat the website of the workshop. Many details have to be defined yet, and the application is not open, but you can regularly check it. So given that, I'd like to introduce, well, to thank Alvaro for being here with us for his last lecture. So whenever you are ready, please share your screen and
okay. I assume everybody can hear me well.
Yes.
Perfect. Okay, so I'm very happy to give you the last of the 3 lectures. And just to summarize what we have been talking about in the last 2, I remind you all that the main question, let me see if I can actually minimize this. Oh, perfect. That the question we have been asking is how reproducible microbial community assembly is. And I've been giving you a few examples from folks who have been studying this question in the wild. For instance, this is the study that looked at the community assembly in the water tanks that form within the foliage of bromeliad plants. And the take-home message from this is that if you look at plants that are very close proximity to one another, it is a natural experiment to study variation. And you find that there is a lot of variation taxonomically at the lowest levels of taxonomy. Fewer than 1% of all OTUs are present in all plants. Yet when you look at larger emergent patterns of metabolic organization across the communities, when you look at the fraction of the metagenome that is devoted to different metabolic pathways like fermentation, respiration, heterotrophy, cellulolysis, etc., you find that there is a very strong convergence across habitats, and that most habitats contain very similar fractions of their metagenome devoted to the same metabolic pathways. And part of what motivates the work we do is that understanding these patterns of convergence at higher levels of organization, this emergent simplicity that occurs at higher levels of organization, despite the large variation that occurs at lower levels, the species level in particular, understanding the ultimate causes of that is very difficult in natural habitats. The main reason is that the community assembly is shaped by both deterministic and stochastic processes, and all of them happen at the same time. And moreover, there's often a large number of unknown unknowns, right? We often do not know what the selective pressures are in these habitats at a quantitative level. We very rarely have a complete picture of the entire assembly history of these communities, of which species arrived and which were not able to fix in the communities. And we often lack a detailed understanding of the environment as it's being modified by the species that are living within. And therefore, it is complicated to address these questions simply by looking at natural environments. So what my lab does is we study these questions in well-controlled habitats where we have a detailed knowledge of the chemical composition, the physical properties of the environment, the nutrient composition, at least the ones we add in. And also because these are liquid communities, it is straightforward to even do analytical work to understand what is the effect that the microbes have on the environment. And we also have control over the ecological processes, the history of colonization, from where migrants come, how connected the habitats are, and you will see an example later of how we can manipulate that to gain understanding. So we can manipulate many of these parameters and understand a lot of what is going on. So that gives us a tool to try to understand what are the rules that give rise to these patterns of emergent simplicity in microbial community assembly at both levels of organization, taxonomic and functional. So let's see. The experimental pipeline, again, so everybody is on the same page. What we've been doing is we do high throughput enrichment communities. We take natural samples, we stick them in water, then we take this goo, and we filter the bacteria out, right? So we get we extract the bacteria that live in here. Again, this is the bacteria that were in the environment. We're not trying to culture them or anything like that. Just take them as they are, and then we throw them. We sample from that very large diverse pool into what you can think of as a little test tube that contains defined synthetic medium. This is, again, for those of you who are familiar with this, M9 plus a given carbon source. And these are carbon limited environments, which is where carbon is the limiting growth factor. And then once we inoculate by sampling randomly from the pool of species in the inoculum, we let the bacteria grow. And we typically incubate for 48 hours. And after 48 hours, we take a random sample of cells from here, and we add them into a new test tube where we have replenished all the nutrients. We allow the bacteria to grow subsequently, and then we apply another bottleneck, and we can repeat this for multiple days. And each time at the end of each growth period, we do community level 16S sequencing to measure the composition of these communities. This is, you've seen this plot many times. This is an example of what population dynamics look like. And as you can see, after 8 to 9 transfers, we find that communities reach an equilibrium where community composition changes very little. And one of the main findings we've made is that even in these very simple habitats, we find a family level convergence, where if you repeat the same experiment from the same inoculum in 8 replicate habitats, what you will find that even as I showed you yesterday in even many more than 8, and I'll show you more today, you find that at the family level of taxonomy, there is a substantial convergence. And all of these habitats contain very similar fractions of the same 2 dominant families. Yet when you look at the species level composition, you find that there is a substantial variation from community to community, despite the fact that we are doing the same experiment multiple times. Okay, so I've organized this series of lectures into three lectures, right? With the first one, I introduced you to this question, and I gave you an overview of what mechanisms could be supporting coexistence in these communities, despite the fact that there's a single limiting resource. On the second one, we addressed the question of why community assembly is so convergent at the family level and how we can explain that. And the answer was that family level convergence reflects a metabolic convergence, and functional what we are seeing, the signal we're picking up by looking at this family level convergence actually is reflecting a functional convergence, a metabolic organization of the communities in 2 guilds. You remember we discussed this a couple of days ago. One of those guilds are respirofermentative bacteria that, when particularly in glucose, which is the case, and other sugars, which is the case we've been focusing mainly, they specialized in the supplied resource, and they grow very strongly on it. And by product of growing so strong in glucose, they employ a form of metabolism that is also wasteful. This reflects a classical trade-off between growth yield and growth rate. and they secrete all of these other nutrients, which are fermentation byproducts, which are acetate, succinate, lactic acid, and so on. And those are being uptaken by the Pseudomonadacea, which we find in our communities. So the fact that those traits are conserved at the species at the family level explains why we see family level convergence, and that was the subject of the lecture I gave a couple of days ago. And today's question, what I wanted to focus on is the final those questions, which is why community assembly is so variable at the species or genus level. And today's lecture, I just I made it a bit shorter so that it would give more time for questions that you may have had, not only about this lecture, but also about the other two. So hopefully I will finish a bit earlier than in the past couple of lectures, right? That's that way by design. All right, so I wanted to start by prefacing this talk by telling you that the result, this idea that there's strong family level conservation, despite substantial genus level variation, species level variation, we focused a lot on glucose because that's the environment that we've attempted to understand better. But this pattern is conserved across the board for all nutrients we've found so far. And here's an example. This is some citrate. Citrate is an organic acid. And these are just two examples from two different inocula, each of which was propagated at different times, right? And here you have the genus level population after equilibrium for the eight replicates. And as you could see, there are various alternative compositions. Yet in all cases, the family level composition is very similar. This is another example where we find that different composition at the genus level again, and across these 8 replicates. Yet when you bin the reads by family, you find again a fairly consistent structure, right? And we have seen this not only for glucose, even though despite the fact that today I'm also going to be focusing on glucose, but I just wanted to tell you that this is not some weird artifact of sugars. We see the same in other nutrients as well. All right, so I'm going to evaluate 2 different hypotheses for why one might see what we're seeing here. Why do you find that there's so much variation at the species level, despite the fact that all these communities converge to very similar compositions at family and functional levels? And I'm going to start from 2 different hypotheses, one of which we proposed ourselves in the original paper where we published this, another one which has been proposed recently. And actually the 2 make a lot of sense. One of those hypotheses is that the reason why we see coexistence of multiple members within the same guild might be due to neutral processes, right? That it could be that all members of the same functional guild might be equivalent, they might have the same fitness, and they're actually coexisting neutrally. So that gives rise to the fact that in some cases you have some members of that guild, and in other cases you see other members of the guild. A second hypothesis is simpler even, right? Is that the reason why we are seeing different species in different habitats is because we are not sampling all species in all habitats. When we inoculate from the regional species pool, we have a large and very diverse community with, even at very shallow sampling, we're seeing thousands of different unique sequence variants. And most of them are at low abundance. So it is plausible that the reason why we are seeing that results like this is because, for instance, it might be that this blue genus, this species was sampled in this well, but not in that well or that well, right? Or that well by that matter, right? Maybe there is a competitive hierarchy, and the only reason why we see that there's different members in different genera, different members of the same family, different genera, on different habitats is simply because they didn't get there in the first place, and it is all due to stochastic sampling at the time of inoculation. So those are 2 plausible hypotheses that might explain why it is that we see alternative states in our communities and we see so much variation. All right, so I just wanted to preface this component, and we're going to test the first hypothesis in terms of neutral coexistence. And I'm going to get back to this hypothesis later on, but for now, I just wanted to tell you a bit of the work we've done to try to understand to what degree coexistence in these glucose habitats is neutral. Right? And what we would expect if we have neutral coexistence is that the frequencies of the 2 species coexisting in the community should remain approximately constant, or at least don't show any pattern relative to the original inoculation frequency that you add them in. For instance, if you take two species together, right, and you mix them in our habitats and you propagate them in pairwise culture, and they were coexisting neutrally, then you would expect that if you inoculate one at high abundance and the other one at low abundance, maybe they're going to remain more or less the way they were, right? There's going to be, if they do have the same fitness, there's going to be very little variation over time, or at least as much variation as you would have simply by purely stochastic processes of dilution and growth. And then, therefore, you would expect a pattern more or less like this, where depending on what was the initial inoculation frequency you have, you're going to have that those frequencies are more or less preserved over time on average. If you have stable coexistence, on the other hand, what you would expect is that it doesn't matter what the initial inoculation is, a frequency is that those communities are going to end up converging to the same equilibrium, right? And there's going to be strong selection to bring the community to a state where the two are found together. In other words, both species can invade each other when rare, right? And that is one of the hallmarks of stable coexistence. So if what we had here was neutral coexistence, you would expect that if we mixed species in pairwise co-culture, we would see patterns that look like this. Whereas if we had primarily stable coexistence between our taxa, we would find that if we mixed the species in pairwise co-culture, they should converge to an equilibrium, and we would observe patterns like this. And I'm going to tell you a little bit about the work we've been doing. I just wanted to highlight that all of this work has been done by Changyu Chang. Changyu is a student in our lab. He's just an amazing scientist and a fantastic person, and we have all been so enriched by his presence in our lab and in our lives. It's been an incredibly rewarding experience to be Changyu's mentor, and I can speak from everyone else in the lab what a terrific influence he's had in all of our scientific endeavors and in our growth as people. All right, so what Changyu has been doing, he has been testing this hypothesis that I was suggesting before, and the idea is very simple, right? What he has done is he has done a really massive amount of work on isolating bacteria from all of our glucose communities, and what Changyu has done is that he has attempted to compete them, right, at different frequencies and see what kind of competition we would observe. For instance, he could take the species, the communities that form in our, the communities that form in our habitats, isolate all the members, right, and then grow all the members in monoculture. Some grow, some don't, right, interestingly. And then the ones that grow, what he would do is he would take them and then mix them at different abundances, right? For instance, he could take this, this is sound like a cartoon, he could take one species, this red species over here, and grow it at high abundance relative to another species, which is this yellow guy here, which is grown at low abundance. Then he could mix the two at equal abundances, or he could mix the two in such a proportion where the red species is rare and the yellow species is common, right? And if the two are growing, if the two are coexisting stably, then you would observe that these three cultures that were started at different initial abundances are going to converge to the same equilibrium. If one of the three were competitively excluding the other, what you would expect is that no matter what the initial frequency of the three species is, they're all going to converge to one of them. And in this case, for instance, the blue community being dominating the red, right? So the red will go extinct in competition with the blue, regardless and no matter of what the initial frequency is. And finally, what we have here is that if you had neutral coexistence, what you would expect is that if you mix the yellow and the blue strains, when the yellow is high abundance, or where the two are the same, or where the yellow is at low abundance and the blue is high, then over time the two would maybe be drifting apart, but on average, right, you would see all of them converging to the same frequency that they had at the beginning, more or less. So to test this hypothesis, what Changyu did is he did exactly this, right? He isolated all of the bacteria in 13 communities, and he wasn't able to isolate all of them. Like a small fraction of those could not grow on their own. They require their partners to be able to grow in these environments. But he was able to isolate most of them, and I think on average it was about 90% of all the taxa. And he did exactly the experiment we were discussing before. He isolates all the members, and then, for instance, here he takes the blue and the red, and then he mixes them at different initial frequencies. And once he mixes those, he would repeat the same experiment that we had done before, right? So we had done assemble these communities from the top down, and what he's now trying to do is, well, if we assemble now from the bottom up, are we going to recapitulate the same behavior? And I'm going to also tell you later some work that we've done to basically the same idea of once we have communities that have been assembled from the top down, we can now reconstitute them from the bottom up and try to use that to understand more mechanistically what is going on. All right, so what Changyu did is once he inoculated these communities, then he, on the same environment where the communities had formed, in M9 plus glucose with 48-hour growth periods following by the same exact dilution factor as we used before, he propagates them for 8 different transfers, right? He inoculates, he lets them grow, and then performs the dilution. He basically treats these communities as if they were the parent community where they came from. These pairwise communities, which is only a subset of the entire community he got, but he propagates them in exactly the same ecological regime as the community where they came from was assembled. And then after that, he uses colony counting to measure the frequencies of the two. And he's been using also some other methods more recently that seem to be in reasonable agreement with the colony counting. All right, so what Cheng has done is he has assembled a large number, around 180 pairs of these competitions, right? By isolating bacteria that grew together and were found together in the same community, right? This is only counting competitions between bacteria that were already coexisting in the same community, but he's now looking at pairwise coexisting of specific pairs. And what he's doing is he's counting the number of occurrences of each pairwise competition outcome, right? You could have, for instance, neutrality, which we've discussed before. You could have a competitive exclusion, which we talked before, or stable coexistence, which we discussed before. It is also possible that you could have mutual exclusion, right? Or frequency-dependent coexistence, which are 2 outcomes that depend on the frequency of the initial that you started with. You're going to end up with one excluding the other, or the other excluding the one, or in some cases where 2 species might be able to coexist when you mix them at high frequencies, but not when you mix them both at lower frequencies. That depends on the exact shape of the dynamical landscape, which we didn't map in its entirety, right? This is just a proxy for what might going on. So, interesting, what we find is that there is extremely few occurrences of pairwise neutral coexistence in our isolates, right? I think there was only 1 case out of 186 where you found 2 strains that are in the same community coexisting neutrally, I mean, having very similar fitness. What this means is that at least in the supplied environment, right, not taking into account how the microbiome changes the environment where they live in, but at least in the environment that we supply, when you strip bacteria from the community context and you measure just the pairwise coexistence, you find that very, very few of them coexist neutrally. And perhaps what is more interesting even than that is that we do observe a fraction of our pairwise coexisting together, but most do not, right? Most pairs that, again, these are pairs of species that coexist in the context of the community, but they do not coexist in the broader context in pairwise, right? So they require the presence of the other bacteria to coexist either stably or in a frequency-dependent manner. And again, very interestingly, we find that neutrality is not observed. So at least in the supplied environment, the bacteria belonging to the same functional guild do not have the same fitness, right? One of them, they clearly do not, unless they, in the case when they eventually coexist stably. All right, so far the idea of whether this might be caused by pairwise neutral coexistence, that doesn't seem to be true, although I will revisit this at the end with some results we're finding that are intriguing and that we are now following up on. So the second hypothesis is whether species level variation across habitats could be due to stochastic sampling from the regional species pool. And again, what this might mean is that some habitats might get sampled some species, whereas other habitats may not, right? So even if there were a species that was belonging to a guild that would be a better competitor than other species, if it is not sampled, it cannot win, right, so to speak, right? So that is perhaps an explanation for why we see alternative states. And the rest of the work I'm going to be telling you today about was done by Silvia Estrella. Sylvie's an incredibly talented postdoctoral scientist in the lab who has done, I think, the most exciting experimental work we've done in my lab so far. And I'm going to be telling you a little bit of the findings that she has made, and this is the subject of a couple of different papers where Sylvie is the leading author. All right, so to ask the question of whether species level variation across replicates could be caused by stochastic sampling from the regional species pool, what Sylvie did is we realized, right, that we only had 8 replicates, and that is really not a lot, right, to really do statistics and try to understand and get to the bottom of to what degree, for instance, stochastic sampling might be responsible as opposed to other sources. So what she did, we also didn't know, like, how many alternative states do we have, right? Do we have, if we repeat the same experiment 8 times and we see, 6 different states, is that, I mean, and if we repeated it 100 times, we find, 60 or 80, right? I mean, we need to know exactly how many states do we have. So to nail down that experiment, what Sylvie did is that she carried out an experiment where we repeated the one we have described before, but on much larger replicates, right? From the same regional species pools of species, we inoculated 100 replicate populations, 100 replicate habitats that are identical from one another. Again, same glucose M9 medium as we've been using in all the experiments I've been telling you about. Yeah, so it's basically the same experiment we've done, but just with 100 replicates instead of 8, right? And only from one regional pool of species. And so Sylvie propagated these communities for 18 different transfers. And let me give you a summary of what she found out, right? So all of these communities, again, under the same conditions, inoculated from the same regional pool, and they ended up converging to a set of different alternative states. And there is some regularity, right, in which states are found, right? For instance, she found, intriguingly, that in some cases, in some set of communities, the only guild that was present above a frequency of 1% were fermenters, right? We didn't find any fermentative bacteria over 1% abundance in those, I think it was about 12 or 13 of them, right? Which is interesting. In all the others, we found a combination of respirators and fermenters that occurred at similar ratios to the ones that I've been telling you about in all these previous talks, right? About 75% Enterobacteriaceae and 25% or fermenters and 25% respirative bacteria. But now, interestingly, what we find is that we do not find convergence at the family level, which is okay, right? Because remember, the family level convergence really is reflecting functional convergence, which is this partitioning of the metabolic space between fermentative bacteria that consume the glucose and respirative bacteria that primarily consume the byproducts. And what we find in this case is that only a small fraction of those where we find a combination of respirators and fermenters contain Pseudomonadaceae, which is this group here, whereas the others do not. And for the others, the respirative group we found is not Pseudomonadaceae, but another respirative bacteria, Alkaligenaceae. And Alkaligenaceae, in fact, cannot even grow on glucose. At least they're known to not be able to respire even carbohydrates as a family. But this one that we have here cannot even grow on its own. This is this orange bar that we show over here. This is Alkaligenes, and Alkaligenes does not even grow, at least this isolate, on glucose on its own, right? It can only grow in the presence of the Enterobacteria with whom it co-occurs. And I will tell you more about that towards the end of this talk. All right, so we find that Alkaligenes can substitute the Pseudomonadaceae, right? But as you can see, you see one or you see the other, right? You do not seem to see the two at high abundances, and this is the key, right? High abundances. I'll tell you more about that in a minute. And interestingly, when you find Alkaligenes instead of Pseudomonas, you find that there are multiple subgroups there, right? You could have only Alkaligenes and this one strain of Klebsiella. This is Klebsiella is we call this KP, and this guy you see over here. And that's it. It's a relatively simple community. But in other cases, right, you could find in this group over here, you could find Alkaligenes as well as 2 different strains of Klebsiella which coexist with it. And you could have this light blue, which we called KM, or dark blue, which we called KP, right? And it is included in some cases the KM it has higher abundance, and in the other cases KP has higher abundance. But in all of these cases over here, there's two members of the fermenter group coexisting with each other, as well as the alkalogens. Now, what happens in these two groups? Well, the same pattern is repeated, but in addition to alkalogens, we also find a member of the Comomonadaceae family, which is Delphtia, which co-occurs with the alkalogens. And in some cases, we also find here a Chromobacter, right? And this is this brown guy over here. It could be found also in some cases here and here in others, right? And interestingly as well, we notice that whenever we find Comomonadaceae, it can only be with alkalogenaceae. We never see it here or here, right? So Comomonadaceae apparently requires the presence of alkalogenaceae in order to be found, right? And now you could have a community where Comomonadaceae and alkalogenaceae coexist with a single strain of the fermentative group, which is this KP strain. But in this group over here, you could have these two groups, alkalogenaceae and Comomonadaceae, coexisting with both members of the fermenter guild, both KM and KP. And in some cases, you even have a few others, as is the case here, where you have another Enterobacteriaceae, which is another ESB, but we haven't really been able to map to any known 16S sequence perfectly. So in sum, what you find is there's a suite of different alternative states, which are easier to see when you do this experiment in much higher throughput. We have like 1, 2, 3, 4, 5, 6 different types of equilibria, depending on the presence or absence of species above 1%. This is the threshold for this classification we're using. And what we notice is that there's very interesting patterns. One is that the first one is that there are some states where there's no respirative bacteria above 1%. This is something we hadn't really seen, or actually we had. There were a couple of communities in the science paper that were like that. We had originally attributed that to just, okay, well, the variation we have. But now we find that it is possible, right? For the respirative group to not even be there, right? And I'll tell you more about what we think that is towards the end of this talk. And we find a bunch of different groups, families that, a bunch of different groups or equilibria that vary in which of the respirative bacteria they have, whether it's Pseudomonas or alkalogens. And we find that when it is alkalogens, it is possible, not necessarily required, that you will find other taxa there, that it is when alkalogens is present, and we find that Delphtia might be there too. And we also find that it is possible for two different members of the fermentative group to coexist. Now, this is very intriguing because in the picture I've been painting so far, which again has been a simplification, is that the fermentative bacteria is feeding this, is cross-feeding this respirative group, right? But here what we're finding is that the identity of the respirative bacterium influences also the composition in the fermentative guild, right? So there clearly is some other process that influences some kind of more bottom-up at the family level process where the respirative group also influences what happens in the fermentative guild, right? And again, I will give you more evidence that is exactly what's happening. Again, that picture I gave you yesterday, and I tried to emphasize it when I was giving the talk, that is a first order effect, and we're pretty sure that dominates this effect of partition between these two functional groups. But we don't need to take it literally, right? I mean, in fact, we know that all of these Enterobacteriaceae can grow on the organic acids, and that they do grow a little, right, during the last 30 hours of the incubation where glucose is not present. It's not like they stop growing altogether. They still can grow a bit, right? So only the primary, the lion's share, if you will, of the glucose goes to the Pseudomonas, whereas the lion's share of the organic acids go to the glucose goes to Enterobacteriaceae, and the organic acids go to the, in this case, Pseudomonadaceae or alkalogenaceae. All right, so if this species level variation that you see across habitats, for instance, the fact that you can, in this case, it could be even different families, right? The taxonomic variation that you observe, if this was due to, caused by stochastic sampling, then you would expect that whenever you see a community that is dominated by alkalogenaceae, then it should contain zero, like it cannot have any Pseudomonas, right? Because again, the hypothesis is that Pseudomonas is not present because it's not sampled, for instance. And vice versa, conversely, whenever you get dominated by pseudomonas, then you cannot have any alkaligenesia, right? Again, if the hypothesis is that you have a state because the other member was not sampled, then it cannot be there, even at very low abundance below 1%. So we would expect, if we plot the fraction of communities that are dominated by P but still contain A below 1%, or are dominated by A but still contain P below 1%, we should expect those to be 0, right, if this hypothesis was correct. And this is what we find, right? When we actually measure it, we see that 67% of the communities that contain where Pseudomonas dominates, I mean 67% of these communities contain alkaligenesia at low abundance below 1%. And I think you can remember the number, it's like 28% of the communities that are dominated by alkaligenesia, all of these communities here, well, 28% of them do have Pseudomonas, and this particular ESB, right? Not just one member randomly of that family, but this particular ESB is present here in a fair number of these communities, although it is present at low abundance. I actually believe it's probably in most of them. Pseudomonas, its abundance here is very close to the limit of detection by our sequencing. So it is possible that it's actually present in far more than that, but we're not picking it up because of we are just not counting enough reads, right? At any rate, the hypothesis of stochastic sampling from the species pool clearly is not correct. And this is the hypothesis that we had originally proposed in the paper we published in 2018. The second hypothesis, whether variability may be due to neutral coexistence, it does not look like it, at least at the pairwise competitions. It's not clear that would work. But as I will point out at the end of the talk, some results that suggest that might be kind of true, possible, at least in some cases, or at least might have some contribution to what we're seeing. But again, we don't believe that this is the primary hypothesis. Now, another possibility, right, that is less extreme than these two, is that we could actually be having a situation where where communities can adopt alternative stable states and where we have multi-stability, right? We see alkali genes at low abundance when Pseudomonas is present, and we see Pseudomonas at low abundance when alkali genes is present, right? So it could be that they are able to persist in the community at low frequency if the other member is present at high frequency. So we wonder if there is a frequency-dependent bistability at the level of the respiratory bacteria that is what is governing the assembly of these communities into alternative stable states. So to give a first pass at this, and again, a very coarse-grained idea was that, okay, can we try to use the Fokker-Planck equation, right, and make the simplest possible model for bistability in this case? And we understand this is actually a complex community, that this is not a one-dimensional system where the Fokker-Planck equation actually might not work, and you could have curls and other situations. But given the fact that the main drivers for community assembly in this system seem to be these two alkaligenesia, pseudomonadesia, maybe it is possible to at least approximate what we're finding through a Fokker-Planck equation. So if you solve it, you can connect the probability distribution with a dynamical landscape, and what that will give you is the location of a tipping point above which, if the abundance of a species crosses it, you will see a transition. If you are here and you force the system, or even by simple fluctuations, the system crosses over, then it will go to another alternative state, right? And the other way around. So that was what we tried to do. And so what Sylvie did was to try to take this idea to heart, and she collected the data she had from the 100x replicate assembly experiment that I've been just describing before, and derived the probability densities of the alkaligenesia and the pseudomonadesia in our communities. We're plotting this in log scale. As you can see, this is the data I was telling you before that dispels the notion of stochastic sampling. Alkaligenesis is present at low abundance in many of the communities where you do not see it above 1%. And Pseudomonas is also present at low abundance the communities where it isn't present about 1%. So what Sylvia did is that she just fitted this data, this histogram, to calculate this probability distribution, right? And from this equation, she was able, from the solution of the state-state solution of the Fokker-Planck equation, she was able to solve the putative potential on which each of those two species is moving. And what this would give us, for instance, would be the location of the tipping point between the two, right? How far could you go? Where if there was a fluctuation that would push a population of Alcaligenesia above this point, would it switch to another state, right? And the same thing for the Pseudomonadesia. And what then she did is that she tracked population dynamics in a range of these communities, and she in particular was recording, I mean, the entire population abundance as a function of time for all the taxa we have. But here I'm plotting the probability, the abundance of alkali genes as a function of time for all the transfers for a collection, I think it was 24 different communities, which we chose representing both states where alkali genes dominated and those where it didn't. And here, this dashed line here represents this point here, and this the shadowed orange area, that's the uncertainty we have in determining where this minimum equilibrium would be, right? And what we find is that by and large, it does seem to be working, right? That once the alkali genes exceeds this region over here, jumps over this threshold, it tends to jump over and goes to this equilibrium. Whereas if it never does, it remains at low abundance. And the same is true for the Pseudomonadesia, right? Once the Pseudomonadesia exceeds this flat region near the tipping point, it does go up and climb up all the way up to this other equilibrium. And if it never does, it might ever drop to this region of low abundance here. So at least this very coarse-grained model is giving us a very good idea where the tipping points in our system are, which we can corroborate through dynamical experiments. So now the question is, okay, so we have assembled these communities from the top down, but one of the things we can do is, as I was saying before, is we could now take it apart and isolate all of these dominant members. We were able to isolate at least the Pseudomonas, this genus here, these two strains of Enterobacteria, and we were able to isolate the alkali genes. We were not able to isolate the Delphtia yet, and indicating that Delphtia clearly requires the presence of probably alkali genes at least in order to grow in our habitats. But we're able to isolate the other three. So we were now doing experiments where we could try to take, and by the way, I told you before, alkali genes doesn't grow on its own in this environment, so it needs the fermenter to be there. So what we did is that we took KP, which is this dominant member of the Enterobacteriaceae family, this dominant fermentative bacteria, and then we would mix, for instance, Pseudomonas and alkali genes together at different abundances, and then that way we could reconstitute our communities. And we would repeat the same experiment as before. Now we grow, and then we propagate for 12 transfers. And after that time, we plate to count the number of CFUs belonging to each, right? So what we were able to do then is we started mixed cultures of Pseudomonas and alkali genes at different initial abundances with the same basal concentration of KP, which is this fermentative bacteria. So this KP is present in all of these communities, and each square represents a specific abundance of initial abundance of Pseudomonas and alkali, just one, right? So it would be here at the center of each of these. For instance, here the abundance of alkali genes would be 10 to the minus 1, and the abundance of Pseudomonas would be 10 to the minus 4. Here the abundance of alkali genes is 10 to the minus 4. The abundance of Pseudomonas is 10 to the minus 3, and so on and so forth. And in all of these, we also have KP present, right? Because again, without KP, you would not have any coexistence. And as a function of time, what we find is that we did this experiment in 2 replicates, and we are coloring each of these squares is orange if the community contains KP plus A both about 1%, It is purple if it contains Kp plus P over 1%. It is blue if it only contains Kp. And it's gray if when we do the replicates, in one case you get one result and in the other you get the other, right? So if in one case you have that A is dominating and in the other you have that P is dominating, or if in both we found both A and P together. So we just, when we cannot tell, we just label them gray, right? And what we find is that as a function of time, this community converges to, this phase portrait converges to a state where every community that was started above in this orange region converts to a state where alkali genes will dominate. And every community that would be started in this purple region would converge to a state where Pseudomonas would dominate, right? And here I am just showing you an example of the temporal dynamics for each of these six wells, so you can see it, right? So for instance, here where which corresponds to this square over here that starts at 10 to the minus 4 alkali genes and 0 Pseudomonas. Not surprisingly, alkali genes just kind of dominates because it cannot be challenged by Pseudomonas. In this one here, you find that Pseudomonas was originally very low abundance, and then over time you find that Pseudomonas actually went away and alkali genes dominated. And here, on the other hand, and these levels over here, where alkali genes was representing these two squares over here, alkali genes started off being low abundance, and then it never were able to invade, and Pseudomonas was present, and the same was true for this other point here, where alkali genes was never there, right? So Pseudomonas in co-culture with Kp remains low abundance, and these results were obtained by plating CFUs, and the state becomes non-invasible. So when we did this, is that we could take this phase portrait, right, and then we were able to obtain, right, the basin of attraction in the simplified three-species consortia. Remember that the Comomonadaceae is not there. None of the rare members of the community are there, and there are quite a few, right? We only have these three members: Kp, which is this Klebsiella, which is the fermented bacteria, then alkali genes and Pseudomonas. And from those bottom-up experiments, we were able to reconstitute the basin of attraction for the two states, which we shade here in purple and orange. And over them, we're overlaying the composition of our communities. This is the top-down assembled communities now, the 96 of them. And you find that there's a group here, which represents the one where alkali genes is present at high abundance and Pseudomonas is present at low abundance. Here are all of the ones where Pseudomonas could not be detected below the limit of detection, which may be because they're not there, or they could be very rare, and we just didn't pick them by sequencing. This other state here represents the state where Pseudomonas is at high abundance and alkali genes is at low abundance, and roughly corresponds with the basin of attraction that we have determined, right? It's within the boundaries of the basin of attraction as we determined by just a bottom-up manner. And we finally have this other state. Oh, maybe I didn't show here. Okay, and we finally have this other state here, which represent those where you might remember that that there were no alkali genes or Pseudomonas above the above 1%, right? So there's only fermentative bacteria and above 1%, and it's this group over here. Now, if you plot the entire dynamics on this phase portrait, which again we inferred from this simplified three-member community, results seem to actually be making a lot of sense. So communities that are started and they get stuck around this region, they kind of wander around this wide area and have a hard time moving over. Whereas those that started here, for instance, or that rapidly, where alkali genes rapidly went over the threshold, rapidly converged to an equilibrium, as you see in these cases. When Pseudomonas, the attraction well of Pseudomonas appear to be more shallow, and that is reflected by the fact that you see a lot of variation when you find a community dominated by Pseudomonas, and you can also get stuck right in this low, what we interpret as being a low slope region of this dynamical landscape. And this is very metaphorical. I would grant you that we haven't really mapped out the full dynamical landscape in a lot of detail, but at least what we find is consistent with that idea, right? That when you get a population close to this kind of separatrix between the two, you get slow dynamics that take a while until they converge to something, or at least it is possible that you can get stuck in there, right? All right, so I wanted to zoom in into this community here of this state where there is no Pseudomonas or alkali genes. So at least they are there, but at low abundance. So again, corresponds to this group over here. Our hypothesis actually, and it goes back to this, right? Our hypothesis is that these guys are stuck, right? Either there is another metastable state that we are not resolving here in our bottom-up community, or more likely it is that these guys are stuck in this very kind of slow region of the dynamical landscape, where it just takes a long time for either alkali genes or Pseudomonas to evade. I mean, if you see in these cases, for instance, they are both there at decent abundances, and we can pick them up by sequencing, even at the end of 18 transfers. It's just that they never quite jumped over, right, where they are supposed to go. So to test that hypothesis, we thought, well, what we could do is we could now connect the habitats, right? So at the end of every transfer, we could pull species from all these wells and then redistribute across all wells, so that if every well received migrants from other wells, it might give them the push they need to jump over this, getting them unstuck from where they are, right? And converge to one of the two alternative states. So that's the experiment we did. We repeated the same experiment. We took an inoculum, and then we inoculated communities, and then we either kept pushing migrants from the regional pool of species, or we pulled all of the communities and then redistributed bacteria across, so that all these habitats are very well connected to one another, and then receiving migrants from all. And then we did that for 12 transfers, and then we allow the communities to stabilize for another 6 transfers afterwards. And the outcome is very encouraging, right? When we did that, right, the communities are connected, then that state that is dominated by Klebsiella KP goes away, right? And then what you find is that alkali genes is able to dominate, and you find them in all the communities. And I would say that intriguingly, we find that all of these states over here, they're all over the separatrix. They all should be converging to an alkali genes dominated state. They just simply never quite made it, right? So this is consistent with this result here, which is when you actually allow for migration, alkali genes is dominating and pushing the system in that direction. And we are still analyzing this data to really understand better what is happening. It is quite intriguing also that once we stop that when we're doing this experiment with global migration, we find also that in all of these states, you find both of the Enterobacteriaceae. So we never see exclusion, right? Further suggesting that maybe pointing out a potential role of neutrality here. And that's what we wanted to test, right? So this fact that you see that when you are connecting all the habitats, then stochastic extinctions are less likely. We wanted to see if that would be something we could observe also from the bottom up, right? So now the first thing we wanted to do is to ask, well, if we mix the two together, the two fermentative bacteria together, are we going to see that they coexist, or are we going to see, as we see in this state, that the dominant one, KP, outcompetes the other? And we have found is that, and no matter what the frequency you mix them, KP always excludes KM. By contrast, we also find that when you mix Pseudomonas with KP and KM, the two fermented bacteria, KP excludes KM, and that is consistent, and we see this here, and that is consistent with the fact that you never see them together when Pseudomonas is present. Yet we find that when we mixed KP and KM with alkali genes, it is possible to, depending on the initial frequencies, in all 3 cases, we did multiple replicates, which we show here, and all these communities, for simplicity, I'm only showing here the ones that were starting at equal abundances, but they all end up coexisting after 6 transfers, and in some cases they can remain around 50-50. In other cases, one goes up, in other cases the other one goes down, and this is consistent with this finding here, where the abundance of one or the other could be quite variable, in our top-down communities. And to further cement this point, we inoculated, here I'm showing you the result of repeating this experiment, this one over here, where KM and KP are, the 3 different replicates are inoculated a high abundance of KM, a half and half, or low abundance of Km and high abundance of Kp, right? These two different Klebsiella strains, that are part of the same guild, are inoculated at different initial abundances. And the result is at least somewhat consistent with a neutralization of their competitive dynamics, which we see is only happening when alkali genes is present. So that led us to hypothesize that perhaps there is this emergent neutrality where the members of the same guild might actually have the dynamic that are finding coexistence in these communities. Their dynamics might be more neutral than you would imagine simply by looking at them in pairwise competition, which again emphasizes the importance of taking the entire ecological context and the difficulties of inferring what's going to happen if you only do pairwise competition. I'm just here showing you some examples of the type of dynamics we find. And we are now trying to really find traces of neutral dynamics among the Klebsiella in these communities to see if we can validate our hypothesis. All right, so wrapping up here, the final question I wanted to address in this series of lectures is why community assembly is so variable at the species and genus level. We have found strong evidence for multistability. We can find very strong evidence against the idea that there is a stochastic sampling is what's governing this. We also find, however, that an evidence, at least, for close to neutrality between members of the same guild in the context of a complex community, which we do not see when we just grow them in pairwise co-culture. And that is it. And this is my final lecture. So I really wanted to take just a couple of minutes to thank the amazing folks in the lab, which is make such a joy well, I don't go to work every morning anymore due to the pandemic, but I still communicate with them every day, and it's just so wonderful to work with all of them. You know, it's just one of the greatest experiences of my life, without doubt. So that's all I had for you. I hope you have enjoyed the lectures and taken something from it. I'm happy to take your questions now.
Thanks a lot, Alvaro, for the very nice lectures. So there is a question from Silvia.
Yes, thank you for the very interesting lecture. I just wanted to ask a clarification. I didn't get the reasonment that you used to exclude stochastic sampling as a cause for the variability. Could you explain?
Yes, of course. So, okay. Well, if it was stochastic sampling, and by that I mean, I'm not saying that it is not possible that, of course there is stochastic sampling, right? What I'm saying is that the reason why we see variation in community assembly is not because a species is not sampled at all, right? We just don't add it to the well, right? Because if it were, right, in these communities where we have Pseudomonadesia, right, if the reason was that you would have seen, imagine that the one hypothesis would be we would have seen alkaligenesia everywhere, and the only reason we don't see it here is because stochastically you didn't add them to those test tubes, right, where you inoculated. Because you would expect to see zero alkaligenesia here, right? And conversely, the alternative would be, well, the only reason why you see no Pseudomonadesia in these communities is because Pseudomonadesia didn't make it. It just didn't get added to the well, right? So in these wells, you added Pseudomonas, but not alkaligenes. And these wells, you added alkaligenes, but no Pseudomonas. And what I am trying to emphasize is that this is visually might be something you could think, but if you examine in more granularity what is the community composition, here I'm only showing the community composition at above 1%, right? But what we find is that of all that in all the communities, this is what we would expect, right? If it was stochastic sampling, right? That if you're dominated by Pseudomonas, you would have no alkaligenes, right? It didn't get sampled, so it cannot be there. Or if you get dominated by alkaligenes, you have no Pseudomonas. Because if the hypothesis would mean that it didn't get sampled, right? But we find that whenever you get dominated by Pseudomonas, in most cases you also have alkaligenes. So both are there, even alkaligenes at low abundance, right? And when you get dominated by alkaligenes, you also have Pseudomonas. Again, both are there. Only alkaligenes is high abundance, and Pseudomonas is low abundance, right? So it's not presence, not presence. It's just presence, low abundance, or the other way around, right? So that rules out that the reason you're seeing alternative states is because a species didn't get sampled, right? It's not saying that could have never have happened, right? It is possible, particularly for some of the more rare bacteria species, that maybe you don't see them in some wells because you didn't get them there. So of course that could play a role, but at least in the dominant members, right, that the ones that we see at high abundance in the end, it isn't really the case that that the reason why we don't see them is because of lack of sampling. I hope that answered your question.
Yes, thank you.
Great. There is a question from Miguel.
Thank you, Jacopo. Truly remarkable stuff, Alvaro. This is a really exciting results. During your previous talk and also during this talk, you have been convincing us that there are some archetypal attractor configurations, and that's reminiscent of this idea of the enterotypes in gut microbiomes. It's clearly in here you actually show this temporal component that is very convincing, but in the case of the enterotypes, there has been quite a lot of discussion about whether or not these are just artifacts, statistical artifacts from compositionality, for example. And I just wanted to ask you if you have an opinion on this discussion from your results.
Right. I am afraid I don't know. I mean, I know the literature, I've read the papers, and I know there's a lot of controversy, but I don't feel like I should be weighing in on it, right? I think it is clearly possible to find, I mean, so on the one hand, it's clear you could find artifactual results simply by lack of sampling, right? So it is possible to see what appear to be, discrete groups of states simply because you are missing the ones in the middle, right? So definitely a big reason why we decided to expand in our experiments to do 100 replicas, not 8, right? Because if you do 8, you might be seeing things that aren't there, right? You might be seeing 2 alternative attractors, but if you do 100, then you, okay, right? Maybe it's a continuum and I'm just imposing categories when they don't exist, right? So that's kind of that, at least I can tell you that controversy really made it clear that we needed to go to higher, to do more replicates, right? And to really understand. And of course, the issue with enterotypes as well is that there's so many, the mechanisms are so poorly, even if they exist, right? That the mechanisms, what are they exactly, right? How do you pin them down, right? And there's all kinds of questions around that too, which beyond the statistical, whether they're statistically correct or not, right? It is unclear, or it's much harder, right, to know if the reason why we're seeing this because the habitats are different, because the regional pools are different. Because I mean, I think there's people doing a lot of work that is really fascinating on trying to elucidate those differences. And I just think that the place I see our work is in trying to create the simplest possible of habitats to at least get intuition of, okay, well, if you were to see enterotypes, right, what would be the mechanism that could lead to them, right? And when would you not see them, right? What would lead to those enterotypes to disappear, right? And that's what we, in some sense, we're after, right? We're developing a ecological understanding of what are the mechanisms that can give you different attractors or not, right? And to give us an intuition for why those might be. But yeah, as for the entire literature, is a really it's a really tricky thing that I'm not an expert on the human gut microbiome. So perhaps I would pass on commenting the specific controversy.
Thank you, Albert.
Great. There is a question from Gabriel.
Hey, really nice talk. I was wondering about the presence of when you're in the alternative state, so when you're dominated by A, P is present at low abundances and vice versa.
Right.
And in particular, I was wondering if potentially maybe phages have something to do with this. So are there phages present in the initial inoculum? And could you do these experiments filtering phages?
Right. This is a great question. So we now have a wonderful phage biologist in the lab who's we are very excited to collaborate with to address some of those issues. I can tell you that we had someone from like a really talented a student from Paul Turner's lab, who's a phage biologist, and came to our lab and tried to find phages that could be grown under the conditions of our communities, right? Basically, phages in the communities, and because these are communities that form in a defined environment, at least we thought that it would be, isolating those phages in the same environment where the community assembled would be easier if they were there. And he tried very hard for a few months and found nothing. So we didn't find any lytic phages going around. We still, I don't know, one question, for instance, could be prophage induction, whether that could have a role, and that we don't know. We have not looked, right? And that could be a very interesting possibility, right? That maybe, just prophage activation might potentially have something to do here with the findings, but at least lytic phages are not around, or at least if they are, we couldn't isolate them. But I agree that this is a really fascinating topic, and we are now trying to understand better what the role of phages could be in community assembly. So this is hopefully in a couple of years or so, we'll have results on that front.
Great. I don't see any more questions. So, there is a question from Tomasa, please.
Oh, sorry. Hi.
Hi.
Thank you very much for the very interesting talks. And I was wondering, one very intriguing thing that you see that is found is that there are a finite amount of stability endpoints in the last experiments that you were explaining. I was wondering, and of course you've done a lot of sampling, a lot of replication. I was wondering if you were expecting a further number of coexistent points, of equilibrium points, whether even a bigger experiment would be done, for example.
Right. That is a fantastic question, right? Like how the number of alternative states scale with the number of replicates you have, right? And yeah, that's a great. We haven't done it, right? And we could try to do some verification curves, I guess, so to speak, right? To find. Once we have the data with hand, we can try to see how often we'll be discovering these states, right? And I think at least that's something we could try. No, that's a great question.
Well, possibly my hypothesis would be that with a simple environment and such finite community, maybe there would be not so many more.
Right.
But yeah, it's very interesting. Also apply to other kind of communities. Yeah.
Right, and I think I was in some sense. Well, I don't know if surprise is the right word, because I didn't have really a prior. It's funny in microbial ecology we don't really have theory. So when we're surprised or not about things, it's often difficult to know why we are surprised or not. Yeah. But at least in this case, I guess the 6 seems a lot for some reason. I don't know why, right? But the fact we have 6 stable states so different from the same experiment seems like, I don't know, naively I wouldn't have expected that many, right? Yes. But I can't tell you why I wouldn't have. It's maybe by induction on previous work. But no, I agree. I think it would be a really interesting question, and of scaling this up instead of 100 to 1,000, which is feasible, right?
No, well, yeah.
It's a bit expensive, but getting cheaper every year. So maybe in a couple of years it'll be as costly as it was when we did this experiment the first time. So but anyway, yeah, no, it will be very interesting, to do that question. Thank you for the question.
Thank you. If I can, if there's no other, if there's time, I have another little questions. It's more of a curiosity. I don't know, Jacopo, what do you think?
Go ahead. There is another question.
Yeah, it's just a curiosity. I found it very intriguing as well, the metabolic convergence to the level of family. And I was as well wondering if you had any expectation for other kind of communities. Let's say I'm thinking if this kind of a relationship would change with the rate of speciation, so with systems where the rate of speciation was different, let's say slower or faster.
If you add migration, because we don't really have speciation over ecological timescales, but we could add species through migration, right?
Yeah.
Right. So I didn't have, this experiment, this is a fantastic question, by the way, and I'm regretting now that I didn't include this slide, because we've repeated this same experiment. In fact, when I talked about this, I think I have this slide here, but I didn't show you the data. Yeah, here. So we've done this experiment where we kept adding migrants from the original pool every single day, right? So we kept pushing species in, right? So we whenever we do the transfers, right, we take an inoculum. So we take the inoculum and add species to it. So we grow for 48 hours. We transfer cells from the old generation into the new, and then we bring migrants from the same inoculum that you used the first day, right? So when we do that, diversity goes up by tenfold. So we're getting like 150 taxa coexisting in our habitats, many of which are at fairly high abundance. So these are not, I don't think these are sink populations. They are, some of them reach like maybe 30%. For instance, we see more axelesia in these communities here. But when we stop the migration, they collapse and they go back to these states, right? So while we're doing this migration, there you have a much higher number of species. So if species keep arriving from the original pool, diversity goes up massively. But there aren't that many alternative states, interestingly, right? And maybe it's because since you are adding these new species, the system has a harder time getting stuck in metastable states. They all have very similar compositions, right? And then when you stop migration, they all converge to very similar equilibria, right? And you have additional families that don't show up here, and then when you stop migration, they go away. So we have done that experiment, and we haven't published it yet. And I think it's very interesting, but we are, it's one of the things that hopefully we'll be able to put out into the world in the next few years.
Thank you very much. Thank you.
You're welcome. Thank you for the questions.
There is one last question from Mathieu that I cut last time, so I feel bad. So Mathieu, please ask the question.
Yes. Thank you very much. I was curious here, if I understand correctly, all the links between species, it's purely competitive. Would it be possible to, what would it change if you now add trophic links to this kind of systems?
So you're saying, I mean, just to qualify your question, there is substantial cross-feeding facilitation. So in addition to competition, there is a lot of metabolic facilitation too, right? So the reason why we see coexistence is because of cross-feeding of metabolic byproducts that the microbes create, right, from the glucose you provide. So that's true. So when you say trophic links, you're thinking, for instance, if there were protists or bacteriophages that eat the bacteria?
Yes.
Right. That is also a really interesting question. Going back to the question Miguel asked before about the phages is something that we are very interested in exploring. Like, what would be the role of, what would phages do to biodiversity, right? Would they promote it? Would they deplete it, right? There are these potential mechanisms like kill the winner, where phages that have a sufficiently large host range might be promoting coexistence because of selective pressure against those that end up dominating, right? So it might be contributing to that. It could also, through lysis, be releasing a lot of metabolites to the environment, which other bacteria could also use for growth. So there's reasons to believe that bacteriophages might promote diversity, right? And as for other predators, I'm less clear, right? If you have protists that are eating bacteria, on the one hand, they might also apply the selective pressures against the more abundant, so that would promote the growth of, at least might prevent those from outcompeting the more rare taxa. But presumably there wouldn't be as much, cross-feeding, because they would be the ones that eat the bacteria, right? They would not just burst it and let it out so that other cells can eat it, right? So this is a really fascinating question, and I think it could be quite easily studied experimentally, right? And it is one of the areas that I would be very interested in moving on in the future.
Thank you.
Great. So I don't see more questions, and we are a little bit late. So I'd like to thank Alvaro again for these very nice lectures, and you all for asking and participating to the lectures. So thanks a lot for being with us. Now at 5 p.m. there should be Justin Ickel, but I propose to take a 5-minute break and come back at 5:05 for Justin talk. If it's okay for Justin. And so we're going to be back in 5 minutes. Thanks again. Great. I think we can close the breakout rooms and we can start in 1 minute when people join back. Okay, I guess people are back from the breakout rooms, so we can start. So the next lecture is by Justin Ekel. So Justin, thanks for being with us, and please feel free to share the screen when you're ready.
Okay, great. Thank you. You can hear me okay?
Yes, perfect.
All right. Okay, thanks again for having me back. Today I want to switch gears a little bit and think more specifically about the energetic dynamics of consumers on landscapes, and think of some of these processes in maybe a little bit of a different way, and then aim to leverage these ideas to examine a really interesting transition in the history of mammalian systems. And this is effectively the evolution of grasslands. And I know you've already had some great talks about the evolution of, or about grasslands and savanna systems, where we are grass specialists. Almost everything we eat is grass. You know, beer is grass juice, bread is grass, rice is grass. We rely a lot on grasses. And so the evolutionary transition when grasslands evolved as a unique biome was a really significant event in mammalian evolution. So that's that's where I'll end today. Okay, let's see. And really what I want to do is look at the dynamics of starvation and recovery, and think of these processes from an allometric perspective as a function of body size. And I want to try to make the argument that this timescale of these processes really structures a lot some of the constraints of mammals that terrestrial mammals experience. And so again, I'm kind of isolating my thought processes towards mammals, and more specifically terrestrial mammals. The rules change a little bit when you don't have to support your own weight, like aquatic mammals don't have to. So starting very simply, right? All organisms, as we all know, must find food, acquire food, and process food. They have to get the energy required for reproduction to pass on their genes to the next generation. And organisms do this in very different ways. Some organisms consume lower quality food that's more evenly distributed. Some organisms consume higher quality food that's somewhat more clumped over space. And other organisms, such as this hyena in the lower left, consume very, very energetically rich foods that are very clumped in space, and also happen to move around space. And these are all unique challenges depending on the different foraging strategy organisms are following. And they all function to distribute risk. So dealing with risk is part of the game. It's part of being alive, right? And organisms evolve different strategies to cope with the risks that they experience. When we think about just finding food, finding resources, we can think about the availability of foods. So organisms must, depending on the foods that they're trying to acquire, they must search for foods on a landscape. And whether that landscape has a lot of food or a little food really has an impact on their strategy. That's in part due to the natural conditions of the landscape, the abiotic conditions. It's also due to the biotic conditions like competition. They have to deal with dispersion, how patchily distributed food is across the landscape. Sometimes encounter rates are very short, and sometimes encounter rates are very long or very large. They have to deal with energy densities, differing energy densities of foods. A lot of foods that are homogeneously distributed tend to be poor nutritional quality. Other foods that are more clumped in space tend to be higher nutritional quality. You know, think on one end of a grazer consuming grass that's relatively poor nutritional quality compared to a carnivore on the other end of the spectrum who's searching for these large packets of food, if we can think of herbivores as packets of food, that are incredibly energy dense, but it takes a lot of effort and skill to find them and track them down and ultimately acquire them. In addition to these food-related risks, organisms at the same time, of course, have to balance the skill of not dying. So they have to avoid predation. And that is going to change across landscapes. It's going to change across, it's going to influence the behavior of organisms. And I would say, to first approximation, many of these strategies to deal with risk can be captured by body size. So body size really alters the risk landscape that organisms experience within a system. And I'll get to that in more detail. I started talking about that a little bit during my last seminar. And we've seen this graph before. Body size is really important. It structures a lot of things in mammalian systems. Here, it's structuring the mortality due to predation. So I'm not going to go through this, explaining this too long, except to say that here we see herbivores arrayed as a function of their mass on the x-axis, and the y-axis is the percent mortality due to predation. And there's this sharp cutoff at 310 kilograms. If you're smaller than that, you are consumed. If you're larger than that, you escape predation. So we know that body size influences interactions, and that is pretty obvious on ecological timescales. However, if we zoom out and think more across evolutionary timescales, I think what is also obvious is that interactions influence body size. And there's this dynamic feedback over evolutionary time between the ecology of an organism as a function of its body size and the selective forces operating on that organism that's going to perhaps shift its body size over evolutionary time. So this is another one of my favorite science figures. So this is in a paper by John Alroy in 1998. I'm looking in the over here, so the upper left, called Cope's Rule and the Dynamics of Body Mass Evolution in North American Fossil Animals, where John Alroy compiled this enormous compendium of body sizes of different North American animals over the last 80 million years, as well as the dates of their occurrence, their best estimates of their occurrence dates. And so that's represented in this upper left figure. Each clade is represented by a line that represents how long it was around. And back here, this is around 66 million years ago. This is when the asteroid hits Earth and dinosaurs go extinct. And we see before that point, and these are just, again, mammals. Before that point, mammals were relatively small. This is a log scale, right? So this is body mass in grams. And after the asteroid impact, dinosaurs are wiped out, at least non-avian dinosaurs are wiped out, and mammals fill the space. There's actually been some really nice diffusion models applied towards understanding how mammalian body size fills this empty space that the asteroid leaves. So they fill this niche space and very quickly attain very large body sizes. You'll also notice there's more structure in this too. There's kind of a gap here that begins to form around 35 million years ago or so, which I'm not really going to touch on today, but there's a lot of interesting structure in the evolution of body size over time. And we call this Cope's Rule, the evolution of larger body size within clades over evolutionary time. And this is observed in terrestrial systems. It's also observed not just for mammals, but across different clades. It's observed in marine realms as well. So Cope's Rule seems to be a very common and large-scale macro-evolutionary phenomenon. Now, another thing that John Alroy did, which was really interesting, is he documented, he took this data and looked at the change in mass over a given period of time relative to the mass of the older species. So he created this kind of a stability diagram, right? Where if the trend is on this side of the graph, and I've kind of rotated it just so the masses line up on the y-axis of both figures, which is kind of a funny way of looking at it. But if the trend is on this side, that means there's growth towards larger body size. And if the trend is on the negative side, that means there's decline in body size within that temporal window of the clade. And what he found is, well, it looks like there's an evolutionary attractor in body size at a lower size, but there's also an attractor at a larger size. And this would describe very well this idea of Cope's rule, where there's this slow tracking towards some kind of attractor at the larger body size limit. More recent work, I think this is a 2010 paper by Felisa Smith et al., and they were looking at maximum body size of different groups. So they looked at carnivores and herbivores. They looked across different continents, and they saw the same pattern, where after the asteroid impact at around 66 million years before present, we have this kind of space-filling dynamic of mammalian groups. Okay, and this on the bottom, I'm just giving you a sense of this large body size space-filling end extremum, I guess. Here is a modern African elephant, and these are two of the largest mammalian species that have lived on land, again, Dinothere in the Miocene and Andricothere in the Oligocene. So Andricothere is about 2.5 times a modern African elephant, so that's pretty big. It's not sauropod size, but it's pretty big. Okay, so today I want to really talk about two things. I want to address two sets of questions. First, what is the influence of starvation recovery on the dynamics of populations? And can it provide insight into processes driving body size evolution? The second theme, or the second set of questions, is I want to ask, how do individuals balance starvation risk against reproductive investment when resource acquisition is uncertain? And what are the evolutionary consequences? And so you'll see how these two different perspectives are really linked together as we go along. Okay, but let's start very simply in something with a model that we're all, I'm sure, quite familiar with, or at least have seen before. But the Lotka-Volterra consumption model, right? So the classic predator-prey model, except that here I've included, saturation of the resource. So as the resource density declines, sorry, I'm getting ahead of myself. So the Lotka-Volterra model really captures starvation implicitly, okay? Because as the resource runs out, the growth of the consumer declines. Because we have this function here, the growth of the consumer is a function of the resource density. So as the resource density becomes lower, the growth of the consumer also is hit. Okay, so I'm illustrating this Lotka-Volterra model on the bottom with this cartoon where the reproductive capacity of the consumer is really tied to the resource density, which I'm just using with these bonsai trees, I guess, supplied by Keynote. Okay, so that's the Lotka-Volterra model. It's capturing implicitly this idea of starvation. But could we perhaps think of ways to better capture the starvation process, explicitly capturing starvation? And could that give us any insight into the dynamics of populations that are really constrained by resource limitation? And the way that we went about doing this was to think really carefully about the timescales of growth and recovery following a starvation process. And so we started with an ontogenetic growth curve. And so now we're not thinking about the population, but we're thinking about the dynamics in the mass of an individual. So an individual organism is born, so it has some birth mass, and then it grows along this this kind of sigmoidal curve until it reaches this asymptotic body mass. Now it will never actually, because it's asymptotic, it never actually reaches the asymptotic mass. So we supply a cutoff. So say at 95% the asymptotic mass, that is what we characterize as an adult size, where the adult is reproductively active. Now, so the adult, its business, it's moving around the landscape looking for food. If it doesn't find food, we have states that the adult can be in. So once the organism hits the adult stage, if it doesn't find food, it starves and it reaches the starved state. So it starves along a different trajectory, and it starves to some point along this ontogenetic growth curve. Now, once it's in a starved state, it can then recover if it finds food again. And it follows another trajectory outlined here, this recovery trajectory, back to its full size. So really what we have here is a two-state consumer model. The consumer can exist in a full state or a starved state. Now, when it reproduces, it takes some time for it to reach its full state. And then once it's in its full state, it can cycle back and forth between a starved state and a full state. To determine the timescales of that process of moving back and forth between a starved state and a full state, we considered the amount of body mass that mammals typically have as fat. And there is a fairly known allometric relationship for the amount of fat mass a mammal has. And again, this is on average. So this is taking the average trend across many different mammalian clades. And I'm showing this, I'm moving my windows around here so I can see it too. But I can see this, or we can see this on the bottom. So we have the amount of fat mass illustrated here by the simple allometric equation where there's F naught times m to the gamma. Now, gamma in this case, as measured from empirical data, is 1.19. So it turns out that the amount of fat an organism can hold for mammals, for terrestrial mammals, is super linear. So an organism that's a mammal that's larger can hold relatively more fat than you'd expect based on its mass compared to a smaller organism. So if we think of this ontogenetic growth curve in terms of the proportion of the adult mass that anywhere along this curve represents, we can actually identify those proportions as a function of the allometric relationships of fat mass and muscle mass. So for example, the proportion of adult mass when you lose all of your fat is given by epsilon sub sigma. So it's 1 minus, this is the proportion of fat mass that an organism is expected to be able to hold given its mass. We can do that for muscle and fat mass as well. And so we call this epsilon sub mu. And so this is just 1 minus this summation between the amount of fat mass and the amount of muscle mass, which has a different allometric scaling coefficient and exponent, divided by the adult mass of the organism. So here we've identified some key points along this ontogenetic growth curve that are going to be helpful for us in determining both what the different states of the organism can be identified as well as the time scales required to move from one state or the other. So we are going to define the full state as epsilon sub lambda. So this is 95% of the adult mass. We're going to identify the starved state as epsilon sub sigma, and this is exaggerated here, it's just a cartoon. So epsilon sub sigma is the mass of an adult minus its fat. So we're assuming, and this is kind of an extreme assumption, but it serves our ends in being relatively simple to calculate. But it's essentially the adult mass without the fat mass. So once you run out of fat, you're in a starved state. But if you run out of fat and muscle, because when organisms run out of fat, they begin catabolizing their muscle. And that's usually the death knell for the organism. But once they burn all of their fat and their muscle, we consider them dead. So the consumer can exist in the full state, starved state, or dead state. Okay, so now we have an explicit change in mass that the organism experiences, whether they're full, starved, or dead, and it doesn't really matter what their mass is if they're dead. So now that we have these allometric relationships for these different rates that characterize how long it takes, the time scale of starvation, the time scale of mortality, which is the process of moving from the adult state through the starved state to the dead state. And again, this is mortality entirely due to starvation. and another relationship for the growth of the consumer, the reproductive capacity of the consumer as a function of body mass. We can look at how these rates change as a function of body mass. So here on the axis, I have the body mass of consumers, mammalian consumers. What we've seen terrestrial mammals obtain is outlined in yellow on along the bottom of the x-axis here. And this is the rate. So this is the average rate of starvation and how it changes as a function of body size, mortality, recovery, and consumer growth. And I just want to point out a couple of fun little features of these rates. And that is these strong upper bounds, these asymptotic kind of mathematical weird things that happen at very, very large body size. So for example, we can solve for the point where an organism is 100% fat. Okay, so given the allometric relationship between the amount of fat a mammalian organism can hold, we can solve for the point at which it's equal to 1, the point at which, at what size would the mammal be expected to be if it was 100% fat? In other words, starvation time at this point is infinitely long, and this is why we have this, you know, this decline here, this sharp decline. Okay, so if we solve for the point where the organism is 100% fat, we have mass is equal to 8.3 times 10 to the 8th grams. And so this is an organism the size of about 140 African elephants, which would be very large indeed. In other words, it would be about 5 times the largest blue whale ever recorded. So that's large, and it's fun, but it's not biologically meaningful. So asymptotes are by definition unrealistic bounds. And of course, if we're thinking about body size evolution, we really require thinking about within lineage selective driver that's also mechanistic. And that's what we want to work towards here. Okay, so we've talked about how these different rates, how we identify different states that a consumer could be in. We've thought about how those rates and timescales change with body size. Now, could we put these into a simple Latka-Volterra type model, but make starvation explicit? So on the top, I have a cartoon that you've already seen that illustrates the classic Latka-Volterra predator-prey model where the reproduction of the consumer is tied to the density of resources. Below, I've illustrated a little bit of a more complicated model where we have 2 states for the consumer. The consumer can be full or the consumer can be hungry. And it moves between the states by starvation, which is illustrated by this red line, red arrow, and then recovery illustrated by this green arrow. Wow. Thank you.
I'm not sure it was about your talk, but.
And there's also rates determining the maintenance of the biological tissues that's being supported by the full and hungry consumers, and they're a little bit different. And so that's illustrated by these orange arrows. The blue arrow here is the mortality. So when you lose all of your fat and all of your muscle, you're kicked out. Now, the other assumption that we make in this model, and I think this is a key assumption, is when you're full, you're full. And so you're thinking about reproduction. So reproduction is turned on when the organism is full, and reproduction occurs at a constant rate. So reproduction occurs at a constant rate when you're full, but then as the organism transitions to being hungry, reproduction is turned off. So reproduction here is a switch. It doesn't scale smoothly with resource density. What does scale smoothly with resource density is the rate of starvation. Okay, so starvation is tied to resource density. As the resources become less dense, starvation picks up. And so in a poor resource landscape, there's going to be more hungry individuals. So reproduction is going to be tuned down. Whereas in a rich resource landscape, there's going to be more full individuals that are reproducing, and which then occur at a constant rate. Okay, so those are the assumptions of the model. I already kind of explained everything on this slide, but I just want to show you the non-dimensional version of this explicit starvation model, where we have the dynamics of the full consumer, the dynamics of the hungry consumer, and the dynamics of the resources. So we have consumer growth being constant, only occurring for full individuals. We have starvation, which moves individuals from the full to hungry class. We have recovery, which moves individuals from the hungry to full class. And then we have mortality of those individuals in the hungry class if they continue to starve. The resources change. They have carrying capacity here normalized to 1. And they're consumed to both replenish. Hungry consumers that replenish full consumers will consume resource at a particular rate, but also hungry and full consumers consume resources just to maintain their own tissues. And that's what's outlined in orange. Okay. Oh, and we called it the nutritional state structured model. And again, all of the time scales are based on this ontogenetic curve. All right. So now that we've discussed the different rates and how they scale allometrically, we've discussed this model that incorporates those various rates that are allometric. We can combine them and we can ask, given allometric starvation recovery rates, can we predict observed mammalian densities? Does this model make any sense? Can we make any prediction to verify that the model is telling us something interesting about the real world? And we think that we can. So the results of this starvation explicit nutritional state structured model map really, really well onto known densities of different mammalian systems. Okay, so the relationship between the steady state densities of mammalian populations as a function of body mass is known as Dammoth's law. I'm sure many of you are familiar with it. We're showing the data here as the blue points. And this is the steady state. This is actually, I think it's individuals per meter squared, but that's fixed in the paper. And we have body mass in grams on the x-axis. The steady states of the nutritional state structured model are shown in green and orange. We have a steady state for the full population and a steady state for the hungry component of the population. When you add these two together, that's the total population, and it doesn't look any different than the green line because again, this is log scale, and so the orange trajectory here is much, much smaller than the green trajectory. But the important thing is that the green trajectory maps really well on top of the data. And so we can modify the intercept by changing alpha in our model, which is the resource growth rate. And when we set the resource growth rate to something that resembles grass, we get this intercept here, okay? And of course, that's going to vary from place to place and area to area. And that might explain, you know, if we were to subdivide these points into different regions and different NPP, you know, or types of areas, then we might find some finer structure here. But really the important take home is that the slope is purely a function of the dynamics and rate equations. And here's just the equation for the steady state down here. Now, as I mentioned, the only source of mortality that we have in this model is starvation. If we were to add an additional external source of mortality, we would modify the steady state trajectories illustrated here. And I'm just going to compare that now. I'm going to now add in an additional external mortality source and see how that modifies F star. What we find is that additional mortality in this model really affects larger species. And you can see that the red line starts veering away from the green line at around 10 to the 4th grams. And so it's these larger species that are really impacted by external sources of mortality. And that coincides pretty well with what we understand about natural systems. When we look at large-scale extinction events, it's often the large organisms that go extinct. If their steady state population sizes are more sensitive to external sources of mortality, as we see in this kind of cartoon view of the world, then that might in part contribute to increased extinction risk. However, there is a lot of ambiguity in the fossil record about whether or not large organisms are more prone to extinction. And part of this is a data problem. There is some evidence that that is the case, that larger organisms have shorter persistence times in the fossil record compared to smaller organisms, but it's really hard to get at and based on resolution limitations of the fossil record. Now, our previous finding was by setting the starvation recovery rate to known allometric relationships. However, we can get additional insight into extinction risk by allowing the starvation rate and the recovery rate to vary. So here we're not constraining the starvation rate and recovery rate. We have starvation rate on the x-axis and recovery rate on the y-axis. And these are simulation results now. So what we're doing is simulating the nutritional state model, state structured model over time, and applying some perturbations to it, just some simulation induced perturbations, and seeing when the system falls below some critical threshold. And we do that many times, and we turn that in. If it falls below a critical threshold, we deem it extinct. If it stays above the critical threshold, then we assume that it's not extinct. And so we can look at the probability of extinction as a function of starvation rate and recovery rate. And what we find is there's two regimes of extinction. So blue is a low probability of extinction, and yellow is a high probability of extinction. And there's really two large extinction regimes. This is actually a Hoff bifurcation, which we can solve for analytically. And so as you move towards this area, you get cycles and cyclic dynamics, and that promotes extinction. Over here, the steady state of the consumer is being lowered too far. So up here in this A regime, we have death by oscillations, and in this B regime, we have death by scarcity. And in the middle, we have this nice little window where extinction risk is low. And what I'm showing in the white dot, and this is for a 100-gram organism, I'm showing the white dot illustrates the expected value of the starvation rate and recovery rate for an organism of 100 grams. Okay, so it falls within this extinction window, or so this low extinction risk window. Now, if we move up to 10 to the 4th grams, we find that the window's changing shape a little bit. It's actually getting smaller. One important thing to notice is that the scales on the axes are changing. And if we move to 10 to the 6 grams, the window is a lot smaller, but the point is still, the expected value of starvation recovery rate is still within that window. Now this window doesn't look a whole lot smaller than this one, but because the axes are changing, it's actually about 30 times smaller. So that's another way to assess, I suppose, the extinction risk of organisms as they increase in size. The window in which their recovery rates and starvation rates operate in a way that shouldn't produce large oscillations in their population sizes is lower. Okay, now, can we say anything about the evolution of large body size? To do so, we ran an experiment, a competition experiment. So the competitive advantage of body size among closely related species is what we wanted to evaluate. We wanted to evaluate if two very closely related species that varied in body mass by a small amount were competing for the same resource, who would win? And we used, classic R-star theory to determine the winner. Whichever consumer pushed its resource to a lower steady state would be declared the winner. So we imagined a competition between a resident consumer that has mass M and a competitor which has mass M prime, which is just modified a little bit. So it's M times 1 plus chi, where chi is the percent change in mass. So the competitor could be a little fatter if chi is greater than 0, or it could be a little leaner if chi is less than 0. And there is a limit because you can only lose so much fat before you dip below that, the amount of fat an organism has at which point it would be assumed to be too starved to continue. So there is a lower threshold to how lean an organism can be as a function of its body mass. So we essentially are searching across different masses of the resident and different values of chi, the percent change in mass of its competitor, to see who pushes its resource to a lower steady state, and see if we can reconstruct that what Alroy was seeing in his data back in 1998, this idea of an attractor at a large body mass. And this is what we found. So let me walk you through this figure. So on the x-axis, we have the resident body mass. This is the mass of the resident consumer. And on the y-axis, we have chi. So above chi, the fatter competitor wins, for lack of a better term. And below chi, the leaner competitor wins. And we're searching across body mass M of the resident and the competitor. So what we find is that for lower body masses, the fatter competitor always wins, and that's illustrated by this blue region up here. At very, very, very large body sizes, the leaner competitor wins. Okay, so it switches. And this switching point occurs right here. This is what we call M-opt. Okay, so let's see, I can't. All right. So this switch occurs at M-opt. And if we calculate the value of M-opt, the optimum mass, so this again would function as an attractor because it's the larger, the fatter organisms that are winning at lower body size, which means that given all other things being equal, the larger organism would be selected for, given this pure resource competition situation. And then at larger masses, it would be the smaller that we're selected for. And the value of that switch point is 1.748 times 10 to the 7th grams. So this is purely a function of the simulation that and the rates that we put into the model. Now, when we look into the fossil record to see the mass of the largest organism, to see how well that maps on to this optimum mass, we find that the observed values, the best estimates for the mass of the largest mammalian organisms are for Andrikotherium 1.5 times 10 to the 7th grams, and for Dinotherium, actually 1.74 times 10 to the 7th grams. So very accurate relative to our simulation. So accurate that we checked and double checked to make sure we weren't in some tautological space that, and it made us nervous for a while, but we weren't. So we were able, with this explicit starvation model, to reconstruct the upper bound of mammalian body size, thereby, putting an expectation, putting a specific model to Alroy's insight from the North American fossil data. Now, we don't have any sign of a lower bound, but our state structured model is pretty minimal. We don't have any higher trophic effects. We don't have anything other than starvation. So we might not expect to see too much from this one simple model. Okay, so I want to change gears now in the time that I have remaining to the second question. How do individuals balance starvation risk against reproductive investment when resource acquisition is uncertain? What are the evolutionary consequences? Okay. All right, so far we have been assuming that only the mean resource density impacts starvation. But of course we know, as I mentioned in the beginning, that resources can be heterogeneous in space. They can be patchy, and this patchiness depends a lot on resource type. It also depends on the consumer body size and the area over which it forages. And we can capture these relationships by the scaling of the variability of the resource and the area over which it forages with body size. And I should mention that this work was led by the postdoctoral Jedi Uttam Bhatt, who's unfortunately no longer in my lab, but he's at UC Santa Cruz. And what I mean by this idea that patchiness scales with body mass of the consumer is, let's consider the savanna landscape. If we look really closely at the savanna mouse, it sees a very different type of landscape in terms of its resource patchiness than does an elephant that's moving across this broader plain. Okay, so in this case, the savanna mouse sees a very patchily distributed landscape in terms of its resources, whereas the elephant would see a much more homogeneously distributed landscape. And we capture this idea that patchiness scales with body size with this parameter zeta. Okay, and this tells us essentially how the coefficient of variation of resources changes with body mass. So when zeta is equal to 1, we have a very uniform landscape, and that uniformity does not change with organismal body size. So as the organismal gets larger and larger, the landscapes gets more and more uniform. Okay, so the coefficient of variation is declining. And sorry, I think I misspoke. The coefficient of variation declines as the organism gets larger. So here, when the organism is small, it sees a more patchy landscape. When it's large, it sees a more uniform landscape. However, when zeta is equal to 2, the granularity of the landscape is preserved as the organism increases in size. So if we consider a smaller carnivore that sees a very patchy landscape, or perhaps a frugivore that's looking for fruit that's patchily distributed, as we get larger and larger body sizes, that patchiness is maintained, for example, by a large carnivore also searching for patchy food. So the coefficient of variation when zeta is equal to 2 is preserved as over consumer body sizes. We simulated a consumer model on top of these spatially implicit areas. Okay, so this whole model is based on this energetic model of consumption, where a consumer has some amount of reserves, and as it finds food, it adds to the reserve. So it's foraging in a landscape, and when it finds food, its energetic stores increase, and then of course they decrease due to metabolic costs. So the organism spends some amount of energy, which we have as B. It's replenished as they find food, and importantly, when the organism's energetic state hits a certain point, it invests a large amount of energy into reproduction, and that energy then is distributed across L offspring in its litter. Okay, so it's a relatively simple model where the landscape and resources themselves are implicit, and what we're tracking is the energetic state of a given consumer. So it actually replicates a lot of the dynamics that we had in the nutritional state structured model, except it also includes the ability for the landscape to be heterogeneous, right? We're not constrained to this mean field type of perspective. So this is really taking an individual perspective of a system and then expanding it to populations. So we can and do treat this as an unconstrained optimization problem and explore different life history strategies over resource richness, variability, and patchiness. But we can also assume these life history traits that I illustrated before, how much energy the organism spends in different processes, how it's distributed across litters of different sizes. We can assume all of those parameters follow expectations from allometric relationships for terrestrial mammals, and then numerically solve for population steady states. Okay, and when we do that, we can find how steady state dynamics of the consumer population changes as a function of resource patchiness. And now this is just kind of a cartoon example to illustrate one of our findings, and that is that when the system is not patchy, we find a single steady state. When it is patchy, or as it becomes more patchy, we have the appearance of an Ali effect. Okay, and the Ali effect is represented by this orange line here, where as we get close to 0, the change in population size over time is negative. And then positive above this critical threshold. I think everyone's probably familiar with Ali effects here. And we can derive a measure of stability of the system by looking at the single eigenvalue of the population. Okay, where the eigenvalue is telling us, giving us the steepness of this trajectory at the steady state, N star, and the steeper the trajectory at the steady state, the smaller the time scale of the perturbations of the system, which implies a fitter system. So we're taking this now as a measure of fitness for the population. When we measure this fitness metric, okay, again, the leading eigenvalue, and where a larger value implies a smaller time scale of perturbations, so a more stable system, we can then map that across body mass of the consumer and how clustered the resource is, which is given by zeta. And so again, 1 is a very uniform landscape, resource landscape, and 2 is a very patchy landscape. And we can see that lambda, or the log of lambda in this case, which is more stable in the blue side, so the larger values are more stable and red is less stable, changes a lot as a function of body mass and resource clustering. And this gives us essentially a fitness landscape or we can interpret this as a fitness landscape where more stable regions, organisms should be moving towards larger body size, right? But it depends on the types of resource and how clustered their resources is in terms of their trajectories towards these larger body size. And there's a lot to kind of think about with a figure like this or a prediction like this. But one of the things that we wanted to do is try to leverage this prediction against something major in the fossil record where resource distributions really shifted. One of these big shifts are really obvious if we look at systems from the Eocene and then the late Miocene. If you look at these 2 illustrations of what the Eocene looked like or likely looked like and what the late Miocene likely looked like, one of the big differences is grass. Grass evolved around 10 million years ago, and Eocene did not have grasslands. There weren't any such thing as grasslands. Grasslands really exploded in the late Miocene. So we might assume then that before the explosion of grasslands, food, especially for smaller mammals, and mammals happened to be smaller before the explosion of grasslands, tended to be more heterogeneously distributed, whereas in the late Miocene, they were more uniformly distributed. So can this perspective that we've built with this really simple model provide any insight into the ecological forces driving the transition to grassland resources? We see evidence of the explosion of grasslands beginning at around 15, but not really picking up until about 10 million years before present. This is from a paper by Caroline Stromberg, and yellow here is the presence of open grass-dominated vegetation in the fossil record. So this is really where grasslands come on the scene. Now with this grassland explosion, this grassland transition, we see the evolution in mammalian diet. So we see many different mammalian clades begin consuming lots of grass resources, whereas before they weren't. These are different mammalian clades illustrated here. And what's on the y-axis is delta C13. So that is a measure of where they're getting their energy. Low values of delta C13 mean they're getting their energy from browse resources, and high values of delta C13 mean they're getting their energy from grass resources, because tropical grasses use a different photosynthetic mechanism, and that has a different isotopic signature than non-grass resources. And on the x-axis, we have age in million years before present. So what you see for equids, bovids, hippos, gomphotheres, and really all taxa, if we average them together, is this move towards grassland resources. It is what we see as things transition from C3 to C4 as grasses become more prevalent. This is in Turkana Basin in Africa. We see the same shift in North America, and it's a general shift in mammalian communities where grasses become available. So we can make the assumption that grasses are evenly distributed, or in pure grasslands, grasses are evenly distributed. And so if you have a delta C13 value associated with grasses, you're getting your resources from a more even landscape. Whereas if you have a lower delta C13 value, that is, that is the value of browse resources, that you'd be getting your resources from a patchier environment. So this is an assumption that we're making when we look at the fossil data. So let's assume then that a homogeneous landscape of grass has a zeta equal to 1, where it's just purely even, which may be unrealistic, but it's going to be close to 1. Whereas a landscape that's patchier is going to have a higher zeta value. What is the zeta value of a patchier landscape? Let's take a savanna woodland that's very, very heterogeneous. We can go and grab pictures of these savanna woodlands from Google Earth. We can turn the vegetation into black pixels and the non-vegetation or the non-browse vegetation into white pixels. And we can actually measure using a box counting algorithm the average zeta of the system. And it turns out if we do this a lot for a lot of different landscapes, the average zeta of these woodland resources is 1.71. So now we have this correlation between high delta C13 values of grasslands, where zeta should be close to 1, and a low delta C13 value of browsers with an average zeta of 1.71. We can then map extinct organisms into this fitness landscape, where again we have zeta on the x-axis, and this is also corresponding to our kind of interpolation of delta C13 values. So we're using the delta C13 values of consumers to map them into this zeta space. And then of course their reconstructed body size. And what we see, if we plot a bunch of different organisms, so we have suids, equids, rhinos, let's just focus on the smaller ones at first, across different time periods. So the darker values are longer ago at around 10 million years before present, and the wider values are towards the present. We see a movement, a trajectory that follows the predicted fitness landscape. As they consume more even foods with the evolution of grasslands, they're increasing in body size following what we would predict based on this fitness landscape. Now things get a little more messy as we get larger and larger and larger. And this is effectively because larger organisms have a lot more fat. Again, fat mass is superlinear with respect to body size. And so they can kind of eat what they want. And so the relationships somewhat fall apart up here, as would be predicted, because they have the, their bodies can take advantage of many different types of resources, whether they're evenly distributed or unevenly distributed. Rhinos are kind of in the middle. They follow the trend, except for black rhinos, which tend to be choosier consumers. They tend to browse for foods, whereas the white rhinos tend to graze. So we find when we look at these extinct species, they tend to follow this fitness landscape as predicted from our very simple model, where we incorporate the energetic demands of consumers and the patchiness of foods. Okay, so thank you. I know that was a lot to fit into an hour, and I appreciate being able to visit Italy, kind of, over the last few days, and thanks very much.
Yes, I hope you enjoy the food. So thanks a lot, Justin, for the very nice lecture. So we have time for a few questions. So please, if you want to ask any, you now know how to do it. Use the raise hand tool or type it in the chat. Well, actually, I have one about the first part. When you have the model with the starvation and grazing, so in some sense, it seems to me intuitive that there is a separation of time scale. Perhaps I'm interpreting wrongly the equation, but that there is a separation of time scales between the starvation versus grazing and the population dynamics. And I mean, what typically happens when you do this separation of time scale is that what emerges like a non-linear functional response. So I was wondering if there is a relation there.
Yeah, that's an interesting question. You're absolutely right. There's a large difference in the timescales, and we haven't really investigated that possibility in terms of how that might be interpreted. So you're saying effectively that you can capture that process, because the timescale of starvation and recovery is so short, you can capture that process with a nonlinear functional response embedded within the pure population equations without the individual dynamics.
Yeah, I mean, for instance, if you have like, let's say, a predator who is in two states, like handling and foraging, what you get is a only type 1 functional response, only type 2 functional response.
Right.
If you do the separation of timescales. So I was wondering if there is a mapping there.
Yeah, no, that's a really interesting question. That would be interesting to poke around with. I honestly haven't even thought about it. So, but that would be interesting, and that would simplify things a lot. I think, you know, what would be really helpful, and I think it's helpful thinking about it in both ways, right? Because the kind of complete picture with all of the timescales allows you to kind of think real hard about the timescales of the different processes, but then how they might condense into a single functional response may allow you to kind of travel to a simpler system with those known timescales. Yeah, that would be interesting. We should look into it.
Yeah. So there is a question from Washington.
Yeah, hi. Thanks very much. Those were really great lectures, and I thought that was really interesting stuff. I just have a quick question. So you apply your model in the context of mammals, and you give this very specific prediction for the upper bound on mammal body size, which matches remarkably with what you get out of known data. What is the difference if you apply this to, say, reptiles or dinosaurs? I mean, what would be different in the model, and have you tried trying to make a prediction there as well?
That's one thing we really want to do. The only thing that, because the model is written generally without assuming any particular exponent or coefficients, etc., we applied those known for mammals because they were readily available, and I tend to think a lot about mammalian systems. My dream is to kind of incorporate a dinosaur perspective. There are some good estimates of, and so basically it wouldn't really take much except a change in the coefficients, and I don't even think, the exponents wouldn't change, but the coefficients would change because, for example, dinosaurs, some were certainly endothermic, some were certainly ectothermic, and a lot looked like they were mesothermic. They were falling in between, kind of like great white sharks are mesotherms, or tuna. And
so it's a metabolic distinction. Basically, the metabolic processes will determine different coefficients, which would give you different.
Exactly. Yeah. So it's really just changing the coefficients of the system. And then maybe the other thing that would need to be thought about is whether, just the productivity of the landscape, and what types of landscapes different, if again, dinosaurs, thinking about dinosaurs, we're foraging within. And I think all of those would be really fun kind of thought experiments to run to see, even if we could, because these are analytical, equation, or, these are equations, we can see like changing what coefficients starts pushing that upper bound up and what coefficients start pushing that upper bound down. Because of course, sauropods push the upper bound up, and, therapsids and other smaller kind of reptile-like mammals had a lower bound. They didn't get as big. So it would be a really interesting kind of thought process to work through kind of the flexibility of this space.
Cool, thanks a lot. I got one other quick question actually, which is, you alluded to the higher extinction rate for larger mass organisms. So I guess I'd always kind of assumed that had to do with longer lifespans and smaller absolute abundances. But it sounds like you're saying there's more to it than that, potentially. Is that correct?
Yeah, I think I think the classic thought, and I don't think it's wrong necessarily, is that especially the gestation times of larger mammals makes it really difficult to recover from any big population problem. I think large mammals, and it kind of falls into this generalist specialist thing that I mentioned in one of the earlier talks. It's like large mammals, they have more resilience within their lifetime. So smaller perturbations to the system within their lifetime. You know, if I don't eat for a week, I'll be unhappy, but I'll survive. But if a mouse doesn't eat for a day or two, it's gone. And so there's more flexibility with those smaller perturbations, but then kind of less flexibility with those large perturbations. And I think a lot of it has to do with gestation time, and just how to how quickly it takes to rebuild those populations.
Cool. Well, anyway, thanks very much for the great lectures.
Yeah, thank you.
Great. So we have time for one quick question from Monday. Monday.
Can you hear me? Thank you.
Yes.
Can you hear me?
Yes.
Yes.
Okay. Thank you very much, Provost, for that wonderful lecture. Please, I want to look deeply into this grassland interaction with the reproductive pattern of mammals. Does this mean that the turn down in the population of mammals can be fast disappearing due to climate change and anthropogenic factors? Because you talked about that the grassland contributes to how fast they grow into big and bigger sizes. So I'm looking at a situation whereby and also affects their reproduction. So I'm looking at a situation whereby if grassland is fast disappearing due to the impact of climate change and other factors, it means we are going to have a lesser population of these mammals. Can that apply?
Yeah, I mean,
thank you.
Yeah, thank you. I think there's a lot of mammalian diversity tied to grass resources. And I would certainly expect as you lose those grass resources, that would contribute to extinctions. I guess I would be careful about the models that I was talking about today. These are kind of like average models applied to like the average mammal. So I would be really careful about making a specific prediction with respect to any specific species based on these dynamics, because they're based on average trends. And each system has idiosyncrasies and a lot more that goes into determining an organism's niche space, et cetera, that would contribute differently to the extinction dynamics. I think, taken across many systems, and this is where these models are more appropriate, where we're comparing average trends across many systems. I think that would absolutely be true. That is, you have organisms that are tied to grasslands. As you take those away, and under really short amounts of time, then we'll witness extinctions of those organisms. I think, one of the important things is in the past, these transitions from more forested or wooded landscapes to grasslands, they were relatively slow. Our grassland indicators in the paleo record, they're over, millions of years, or at the least hundreds of thousands of years. So there was time for evolutionary response. There was time for selection to modify what organisms were doing, which we see when we look at the isotopes. They switch to grassland resources. We're making modifications to the landscapes on timescales much shorter than that. And so I would expect that just the differences in timescales of disturbance versus, the evolutionary process is going to increase extinctions relative to, what you might expect during a slow transition.
Great.
Yeah, thank you. Thank you very much. Thank you.
Yeah, thank you.
Terrific. So thank you very much, Justin, for the lectures and answering all the questions. We are now taking a 6-minute break, and we'll be back at 6:15 p.m. Italian time with Justin again and other panelists for the last session of the school. Thank you very much to everyone. We'll see you back in 5 minutes.
Thanks, everyone.
Okay, welcome back. I see almost all the panelists, so let's wait a few more seconds to allow them to join back, or to join for today's roundtable. Okay, I think, yes, I think everyone is here. So thank you everyone for being back to this last session of the school. I just counted the number of sessions we have, and believe it or not, this is session number 52. So I think that this was a long and exciting marathon, and we are now in the very last kilometer of this marathon. So this session is a round table with many panelists that you had the opportunity to know in the last 3 weeks. So it's my pleasure to welcome again Stefano Lesina, Daniel Fischer, Jonathan Levin, Matteo Marsili, Mercedes Pascual, Daniel Segre, and Justin Icke. So thank you very much for giving the lectures and for being here to this roundtable. So the way I'd like to organize this roundtable is to try to have a sort of a spontaneous and self-sustained discussion among panelists, and also including you participants. So before we start, just a few engagement rules. So if you are a participant and if you are following from YouTube, you can post a question in the chat, and I'll try to find the time to redirect them to the panelists. If you are on Zoom, as usual, now you know it very well, you can either type the question in the chat or use the raise hand feature. Also, the panelists, if you want to say something, just, and I don't give you the word, just use the raise hand tool or write me in the chat. So the title of this roundtable is Going Big, Challenges and Opportunities in High-Dimensional Ecosystems. And this title, without hiding it in plain sight, is an interpolation of the title of the chapters that some of the panelists have written in this book here, which is Unsolved Problems in Ecology, and an interpolation also between the lectures of this school. So the two keywords, in my opinion, that naturally emerge from the lectures and the chapters of the book are coexistence. So the why and the how of species coexistence in large communities. So in some sense, the most fundamental question of ecology, because it's basically asking why ecology exists in the first place. And the other word is dimension, the fact that in large community there is a lot of variation on multiple axes, including many species, many conditions, many traits, many functions. So the fact that in the title we talk about going big and high dimensional strongly suggests that much of the intuition about coexistence and ecological dynamics comes instead from very low dimensional theories and models, for instance, two-species competition. And in some sense, in the title is also implied that this intuition should fail when we move to high-dimensional ecosystems, right? So I'd like to have a first round of answers from all the past panelists about the following questions. So do you think that our intuition that we shaped with theories and experiment on low-dimensional ecosystem should fail when we move to high-dimensional ecosystem, whatever that means? And if you think that that should fail, what intuitions about the low-dimensional ecosystem is wrong for high-dimensional one? So I will proceed with this round of answer in alphabetical orders, like at school. So and starting with Stefano.
Yes, so it is obvious even to a theoretician that no place in the world contains 1, 2, or 3 species, right? And yet, if you take like a book of theoretical ecology, 95% of the material is dedicated to the case of 1, 2, or 3 species. So besides this obvious fact, right, that there are very, very many species in the world, like in tropical rainforest, we can find 10,000 species of trees. What strikes me as very interesting is whether we can use exactly the same tools that we use for small scale ecosystem, imagine like experiments in a laboratory with 1 or 2 or 3 species, to understand what happens when we have 10,000 species. My career basically was a bet on the fact that we cannot really extend the same like type of theories that we did for like small dimensional system to large dimensional systems. And we have clear cases where our intuition fails when we move that to more than a handful of species. For example, like the celebrated rule of intraspecific being larger than interspecific competition in two-dimensional systems in Lotka-Volterra does not hold when n is greater than 2, generally.
Great. So, Daniel Fisher.
Well, I guess as implied by the title of my series, I think the answer is clearly clearly yes. I mean, I think in many ways the most dramatic examples are not having many species that coexist, but having enormous number of strains which coexist and compete directly with each other. And almost any bacterial system that has been looked at, one finds a huge number of strains really coexisting in space and time. And trying to understand that in terms of thinking in terms of niches, that somehow one starts from a picture where each type interacts more strongly with its own type than it does with others, which is a lot of the beginnings of a lot of the work on high-dimensional ecosystems. I think that's sort of fundamentally wrong, unless that is really an evolved characteristic of the system that somehow drives it towards towards that. So I think at this point, I mean, certainly having worked on it, a limited amount, my intuition is really poor as to the things that can happen in high dimensions. I just want to actually mention just one example from recent work on evolution of microbes that really sort of shows how different it can be. So the traditional view is that you can get mostly neutral drift and occasionally get a selective sweep. But the other alternative is just it's always evolving. The populations are always evolving, even without much diversity driven by ecological interactions. And the picture emerges is very different. And there's something, the predictions for statistics of diversity and so on are very different. It took a long time to get to the point where we had enough theoretical understanding to start saying which predictions might be somewhat universal, not depend on all the details, but we do have some understanding of that. And so I think it's going to take a while with the ecology to know, what ways one might try to make contact with data, or which things one should find surprising, which things one shouldn't find surprising, and so on. But I think Phil Anderson's more is different absolutely applies here, and it really needs developing different intuition and different methods.
Great. So Jonathan.
Sure. So I think the question is, as you say, sort of a leading one. But I think that it's worth sort of dissecting the question into two levels of organization. And one is to say, while certainly nature around us is quite diverse, the question might be rephrased as, is it fair to say that we can understand that diversity as some sort of simple combination of what are ultimately fundamentally pairwise interactions? So while the individual Lotka-Volterra rules between two species are not going to change in a many species system, the generalizations we have about what it takes to get coexistence intra and inter might collapse, but our overall framework is still okay. I think the more fundamental question is really, when species are in these more diverse systems, do we really experience what some are calling higher order interactions, where you really have the simultaneous interaction of multiple species impacting a third and fourth and so on? And I think the jury is still out on that one. I think it's easy to answer the first question and say, yeah, if you string together a bunch of pairwise interactions, you can't use the simple rules that you develop from simple, pairwise ecology to understand dynamics. But but I think we'll get over that. I think we'll figure that out eventually. I don't find it as sort of an insurmountable question. But what is really challenging is to say, is even the notion that we should be able to understand diverse communities as these sort of combinations of pairs is something we don't know. And you could even, another way of saying is obviously these models are meant sort of to abstract some underlying mechanistic interaction between species, whether it's through resources or consumers or allelopathic chemicals, whatever you like. And the question might be, when you think about those mechanisms, are those mechanisms one which you think can be described in this pairwise fashion or ultimately require these more complex structures. And I think we don't know enough about nature and about how those methods are really driving dynamics in nature to even answer that question. But that might be the pathway that I would try to kind of veer towards getting some progress on the question at that harder level, is what I might say.
Great. So, Matteo?
Okay, so first of all, well, I think I should say from the personal experience, the situation where your intuition fails is a very fortunate one because it means that really there is something fundamental to be understood. But there are two sides of this question. One is our intuition about models. I mean, when we study a very complex model and whether what we expect is what actually turns out. And the other thing is intuition about a real system. So I will not talk about real systems because I've always been dealing with models. But I have an experience with models where essentially my intuition was right 0.5% of the time. You could flip a coin, and in the end, the answer, the true answer was either yes, your intuition is right, or no. I mean, and these type of models are precisely of the type that many people have been describing. Many people are now describing in this community, and when my work was more related to models of financial markets and minority games, but these are essentially resource disordered system, very heterogeneous systems with many different agents, essentially living on many resources. Of course, the models now that are being studied by Danny and others are more complicated because there is also chaos. But these are the type of models that, in my experience, show you really teach you something. They can really teach you a new paradigm, a new type of how you should think in high dimensions. And I think from what I saw in this school, I see remarkable advances. A very exciting time to be studying ecological systems and high dimensional models in ecology, because I see models and ecology and experiments really coming much closer together than in the past.
Great. Mercedes?
Yes, thank you. Yes, it has been quite an exciting course, and also one that gives rise to many questions and ideas. So on the question you just asked, I would say yes and no. Yes, because I think many of the models we have had in the past do not help us with the very high dimensional systems, but they provide a basis to organize some ideas and some concepts. And I think we heard about that, for example, in the context of the stabilization and equalization forces. There are very good reviews by Saavedra, by Barabás, by D'Andrea, showing that it's highly non-trivial to extend those ideas, and we don't know how to do it to high dimensions. But yet, I think the organization of, for example, the different kinds of traits that we have to consider, that sort of low-dimensional theory illustrated, was very useful. On a different point, I think I like to think, of course, about assembly in this eco-evolutionary perspective. And there, I think the question of when we say high, I think there are some discontinuities, and that what motivates our studies, I mean, Daniel just mentioned the very interesting, of course, microbial systems with the strain diversity. But if we move from the microbial world to the systems that really have motivated us to look at diversity, the rainforest, the coral reefs, we are looking at hyper-diverse systems. And if you add the intra-specific variation, it's enormous. And the network of interactions is enormous. And I just like to think, when I think about pathogens, and I call them in my lecture hyperdiverse, I like to emphasize that biogeography has done the experiments of assembly. And if you take certain pathogens where there is very high transmission, they are completely different in diversity than when you have low transmission. And I'm talking about continental differences and discontinuity in the differences. And when I say that, I say that because I think I will make, I think, the most important, or to me, the most interesting point at this moment. I'm deeply convinced, and maybe I'm wrong, but I'm deeply convinced at the moment that we cannot build hyperdiverse systems at one level of organization without the very high dimensionality and diversity at the lower level from which we build it. So I think like the diversity of the traits and the genetic variation that underlies the ecological interactions has to also be very large in places where we assemble a large number of strains or species. And this is not trivial, because then it leads to the question of what maintains this high diversity of the building blocks. And I think it's fundamental, because I showed you an example in malaria where we know that if we drive the system down in transmission, there is actually a threshold at which we lose the ability of maintaining the building blocks. And I think that when we go from very diverse to the more trivial low transmission systems, we are crossing a threshold of that kind. And so I'm saying it's hyper diverse at different levels of organization. And this is not by chance. It's because the same force, the same selection is operating at different levels of organization. So I think that if that is true, we need to understand it, because it's something very different for very hyper diverse systems that truly makes a distinction with the ones that just exist in below this threshold. And anyhow, that is my pitch here. I think those systems are truly distinct.
Yes, very clear. And Daniel Segre.
Okay, so I'll start by saying that, I mean, the kind of intuition I want to bring forward is something that guides the way I think about this, but also fails at higher dimensionality, has to do with metabolism in microbial communities. And in particular, sometimes you may find that if an organism is very good at using a carbon source, another is very good at using the nitrogen source, then you can think of the complementarities that arise in communities. But of course, the molecular complexity in real environments is much more complicated than this, and there are a number of molecules that contain the different elements and microbes that are very good at using this resource or that resource, and this economy of the molecules becomes really complicated. And I think one of the challenges that I see is that on one hand, I think it's valuable and interesting to build detailed models of each individual organisms and try to push that to the boundary of higher and higher complexity. But of course, at some point, this becomes too much because we don't have enough information. It's challenging in many ways. So the question, I think, is how do we find a good balance between the detailed models where we know how the metabolism of each individual organism works, and the more distribution-based models where we try to capture the general properties of the how different microbes interact with each other. And I think that this is crucial because I believe that metabolism can be, first of all, as I said, very complex, give rise to higher order interactions, but there is also the fact that when we want to study individual communities for all the different applications that microbiome research has, it could be this strain rather than that strain that makes you sick, or this strain that can really produce the oxygen that can change the atmosphere of a planet. So the details matter for a lot of, the reasons we care about communities, the details matter. So I think it's important to strike a good balance between the statistical properties of communities and detailed knowledge of metabolism. And the other thing that I want to say, even if sometimes I like to hope that metabolism is really the driving force of a lot of these communities, and I think it is in many ways, but there are many other ways in which microbes interact that I think are so complicated and are barely taken into account into models, from signals and quorum sensing, contact interactions, the interactions that depends on space and time. So I think this is all an unexplored territory in terms of figuring out how, whether and how to take all of that into account. And I'll stop here.
Great. So it's now Justin, and then.
Yeah, no, I guess I would just say that, a lot of the model approaches that we take are designed to ask very specific questions, and we have to simplify most of reality into these toys to be able to say something straightforward or to understand the models themselves, which can already sometimes be ununderstandable, as Matteo was saying. And I think that clouds, that allows us to point in very specific directions and aim these tools to answer very specific questions, but it cuts out the reality of systems. The fact that there really isn't anything as we put these species into a node, or we say this node represents a species, but there's just individuals, and there's within the individuals there's genomes, and they're all interacting in various ways over very complex environments that are ever changing. We might look at systems where we're considering only the biotic interactions between species, but and those are complex enough. But then we think about the feedback that biotic organisms, the biotic side of the equation has with the abiotic side of the equation, and that they're different within species, but also within individuals in terms of having an impact over both ecological and evolutionary time. So I think it's perhaps unsurprising that our models fall short. But as you add more dimensionality to some of the problems, it really expands the possibilities. And I often, think back, when we're thinking about organisms living in these diverse communities, some of the first diverse communities, we go back to the Cambrian explosion. What led to that explosion? What was it like before the explosion, and what was the kind of instigator of the explosion? Was it the evolution of eyes and suddenly opening this entirely new kind of sensory experience of the world that allows organisms to move through over in terms of their evolutionary trajectories? I had a conversation with Charles Marshall at Berkeley, and he described this process as billowing lava, underneath the ocean, like this changing niche space, ever kind of expanding and evolving with the organisms that are using it. And I guess to me that gives a sense of near infinite combinations of interactions that lead to this diversity. And again, I guess the last thing I'll say is that when we look at these model systems, we make these assumptions often of asymptotic time horizons and stability, but these systems are getting hit from every side all the time. They're in a constant state of flux and change, and they often disobey the very assumptions that we're making to be able to say something and anything about very specific questions that the models are appropriate for, but inappropriate for others.
Thanks, Justin. There is a comment from Daniel Fisher.
Yeah, I just wanted to comment on various aspects. Firstly, I mean, I certainly resonate very much both with what Mercedes said about the being not strictly being a threshold for complexity, but somehow when things get complexity enough, there may be some sort of runaway to getting more complex. And if they're not, they're sort of stuck. And this maybe also relates to Justin and things early on. Just another just comment about intuition from statistical physics and what it might tell one and what it doesn't. So I guess I would disagree with Jonathan. My instinct is that things like whether there are 3 species interactions or 2 species is something which is one of the details, can change things, but it sort of won't make things somehow qualitatively different. But I think there are some things where the intuition from the physics are very bad, and that's particularly the sort of instabilities of systems to sort of runaway event, right? A pathogen that comes in, a new species that comes in, does very well, changes things dramatically. And then I think, and Justin's comments about the crucial bits of being the whole system being very much dynamic, rather than thinking of it as being sort of quasi-static. And then the other bit, and this is relevant for coral reefs, but even more dramatically from cyanobacteria, that one organism having a very big effect, or a small evolutionary change in one organism, a seemingly small one, have enormous effects on everything. And so somehow, even if one wants to abstract out quite a lot of the details, there's still going to be some sort of dominant things and dominant interactions and so on. And I think, again, I find some of those ones are some of the things that are hard coming from physics to get a grip of. I mean, some of the details clearly matter, but I think the hope is that a lot of them don't, if one knows what questions to ask, right? And if one defines sort of the kinds of questions as being ones where you can apply them to many systems without having to redevelop everything. I mean, those kind of questions, I think one has reason for optimism that not everything will depend on all of the details.
Yeah, Jonathan, you want to comment on that?
I mean, I think my comment, is more a question of an empirical one than one that maybe is about sort of what is possible theoretically. I agree with Daniel that I think that it might not fundamentally change the rules. We just might have to have some different expectations. But I guess what I really meant to communicate was just that, when we try and understand nature, the question might be, how much do we need to actually incorporate these interactions that only emerge in diverse systems? And I just think the answer to that is unknown. Whether incorporating them fundamentally changes the way we think about species diversity in complex systems, I wouldn't want to propose at this point, partly because we don't even know what those rules are. And I can tell you that, for example, like I have a PhD student that's currently working on questions about, what even are our expectations for how a 3-way interaction or a higher interaction might modulate the dynamics of coexistence. And you get some really funny things. Now, whether they're any funnier than they would be if you had stringing together a bunch of pairwise interactions, I don't know if that's fair to say, but it's certainly something that violates what we have from the simple 2-species ecology that we focus on. So I think I basically agree with Daniel that I'm not sure there's actually a real sort of game change here. The question might be just how much do we have to incorporate these dynamics or these interactions if we want to understand the dynamics of diverse systems.
I think it could be now to turn it around and agree with what you said earlier, is it could be that there are some of these more complex interactions that are crucial, that somehow sort of drive the environment that everyone else is feeling and everyone else is riding on top of. And so I think in that sense, it's some of the things which in certain ways it could end up being pretty crucial for a particular system, and could also be towards this driving towards sort of getting enough complexity that everything else can somehow complexify on top of that, diversify on top of that.
Yes, Mercedes wanted to say something.
Yes, I'm this whole issue of the dominant processes and that we may hope to understand with models is very related also, I think, with the issue of what can we hope to understand with models in these very, very complex systems. And in my view, probably what I would call progress is if we can not really hope to understand these interactions in any bottom-up fashion that will be credible, because I think it's an impossible goal. I think ultimately, if we can, for example, reject neutral theory and understand that we may be in a situation of niche construction that is the complete opposite, then I would ask, then how do we look at data to know this? How do we look at data to know how far we are from a threshold where we will not lose demography, but we will lose diversity? Those are questions that I think we can approach without understanding the mechanisms. We need to understand which macroscopic properties give away the dominant forces. And what does that tell us? I think anything else is hopeless. I think we will never understand the interactions that have been built to maintain a rainforest. I truly think we will never understand them. But I think we will understand the consequences. We will understand the phenomenology. And I say this because I really like a paper by Mark Lipschitz, Pamela Martinez, and others, where they see that they can predict the strains that respond to vaccination with sort of considering the frequencies and certain forces that indicate that of the order of 3,000 or 4,000 non-core genes are involved in this. So what interactions are they creating? They are not even essential genes. They are not even the genes that define the strains, right? So they must be involved in a myriad interactions. If we look at 3,000 non-essential genes, and they give us some predictive ability just on the basis of frequencies. I think, anyhow, I just think that we have to return to this question that, of course, neutral theory, the main thing of neutral theory was to show that the macroscopic patterns we were stuck trying to study didn't tell us anything. They were not informative. So the question is, which patterns are informative? And I think models will be very useful for this.
So there is, thank you, Mercedes. There is a comment from Matteo.
Yeah, so if I may, I think this is a very important point because trying to understand, say, stylized facts in terms of systematic deviation from neutral theories, I think, because, I mean, my way of thinking of neutral theory is like a maximum entropy model. So a model that where essentially you cannot hope to learn anything about the microscopic interactions from the macroscopic behavior. So I really think this approach to address, I mean, as Danny was saying, I mean, how do you find what is the right question? I think this is a general approach that could be useful.
Yes, one question I guess is whether neutral theory is the sort of a useful baseline for understanding this high-dimensional system. I don't know if anyone wants to comment on this or on either other parts of what has been said.
I think it's only useful in the sense that one of the questions is why do things end up looking that way, but they almost certainly won't look that way once one starts looking at the dynamics. But there are rather too many things that can give rise to distributions that look roughly neutral like. And roughly speaking, it's like assuming that all of physics is like an ideal gas, which is clearly isn't right, but it's somehow useful to make the observations and start looking at how things, what aspects go wrong.
But I use the word neutral theory in a broad sense. So if you are interested in asking, I have a bunch of data on this very complex system, how should I look at it? To ask not whether a process occurs, many processes occur, whether a process is dominant or important, even just important, then I need a neutral model where that is the appropriate neutral model for that question, for that process. And so.
But you mean you need a null model?
Yeah. I'm using it.
No.
Thank you for the difference.
I agree. No,
that's absolutely. But I think like we can make progress that way because we don't even know. Look, the clusters that are predicted from stabilizing interactions, they disappear in ecoevolutionary models at very high dimensions. So my point is, if we ask what should we even be looking at in data to say that process is important, we don't know, but I think the models are useful there. Yes, with the appropriate null model.
So Stefano wanted to say something?
Yes, like I agree with like Daniel on the distinction between neutral model and null model. And I think like that an underexplored null model that is not neutral would be a model with random interactions as opposed to no interactions. And my belief is that by and large, they would produce the same results as a purely neutral model, but removing like the need for the strong assumptions.
Jonathan?
Yeah, so I think Mercedes raises some very good points, and the conversation is going in a certain very pragmatic direction, I might say. But I think the challenge, and especially a challenge for theoreticians as an empiricist primarily, is to say, well, you know, it may be that the only source of progress with these hyperdiverse systems involves some sort of null model statistical physics kind of approach. But the problem is that many people really want to know why that tropical forest is diverse for some practical, you know, reason for managing that forest and so on. And the question might be, how do we deal with that tension between what is potentially the most rational quantitative way forward in a system with many dynamical processes that are maintaining coexistence at some abstract level, and the fact that someone wants to know, are the monkeys dispersing the seeds a critical component to the diversity that I observe in that forest? And you can't really answer that with a distribution or a null model. So I think there's a real tension within ecology about what we're actually aiming for within this understanding of diversity of these hyper diverse systems. And the question is, you know, what is maybe the right approach, or is there a meeting of the minds on that kind of framework? Is what I might pose.
Yeah, but I think there is, I'm with you, but if you mention the monkeys distributing the seeds, also you are thinking about some of the effects of the negative enemies that you can find in the major sort of stabilizing forces. Now, there is a more critical question. You may come to the conclusion, and the most practical conclusion, that if we that given the niche construction that is involved in maintaining that forest, the best we can do is not preserve the monkey, is do what E.O. Wilson is saying, preserve, you know, parts of the landscape for nature. I think once you get to the point where you say these complex interactions underlie hyperdiversity, the only way to maintain it is to give it space and leave it working at that, because these thresholds of hypercomplexity, you are going to lose them if you don't give those systems enough, I'm going to call it abstractly space, enough to essentially maintain this diversity and this level of complexity. And I think the interactions are so rich and have been built over such long time with pieces that are built from eons of evolution, not the species, the pieces that constitute them. That if we cross the thresholds to keep that diversity, we lose it all. I think the only practical solution, one of the components of a practical solution, is not to ask, is this particular thing important? Is like, how do we save the system as a whole, enough of the system as a whole? And I think that the 50-50 idea of E.O. Wilson, whether it needs to be 50-50, but I think there is something very real there and very urgent.
Go, Stefano, yes?
Yeah, I agree with Mercedes on the practical aspect, but I think that this is really like raising the white flag to some extent, right? Like, so we're going back to a sense of like conservation, in which we go back like to ancient texts, and they say, this forest is sacred, no one can enter, or only like the warriors can hunt in this forest, or only like this period of the year we can hunt in this forest. Which is like saying, because we don't really understand what is important, what is not important. We just have to conserve the whole.
No, I completely disagree. I'm saying that when we get to understand, it will be too late, because we will understand that the only way to preserve it is to leave it to, and I'm not saying pristine and don't touch it, but I'm saying you are going to, if you lose what maintains hypercomplexity, and I'm saying, I think we have enough in theory, in the theories that exist, to begin to see that this is the reason. It's not a white flag. It's exactly the opposite. It's the realization of the very rich niche construction that goes on in these systems, and how you can lose it, right?
I agree on the practical aspects. I disagree on the interpretation, but I agree with you that that's the best picture.
But it's not a white flag, and it's not going back to the past. It's like, what is the theory tell us today?
So on this issue about the pieces of the interaction and the building blocks, I'd like to hear from Daniel Segre and regarding the building blocks of many interactions, for instance, in bacteria, and from Justin about the fact that then these building blocks of the interaction sometimes have like sort of underlying regularities and that emerge through scaling. So Daniel?
One thing I can say is, right, metabolism is what it is, right? It is a well-defined system. It's the outcome of 4 billion years of evolution, and I think there are two sides of this. One is that because of its underlying structure, it shapes the way microbes sense their environment and respond to their environment in a very specific way. And I think that maybe, and I really truly don't know, I'm curious of whether ultimately we'll find that if you could build alternative metabolisms, the rules of the way communities work would be similar. But I think that it is important, and there may be aspects of this that strongly depend on the specific structure of metabolism, the way it works. And the other aspect of this is that, which I think also is somewhere in between the extreme statistical nature of complexity and the extreme detailed one, which is that there are classes of bacteria and microbes that have specific types of metabolism. So maybe we will not need to understand the metabolic capabilities of each individual organism, but maybe having a categorization of guilds or types of metabolic activities might be helpful. And it's also true that people keep discovering new metabolic pathways, new capabilities that were not known before. Things that seem quite amazing, like new pathways for utilization of organic acids, and people engineer new pathways. There are newly engineered pathways for carbon fixation. So looking also forward at this rising field of synthetic ecology, where we can try and build new communities based on what we know, I think striking a good balance between detail and the statistical nature of communities would be important. I don't know if this addresses your question, Jacopo.
Yeah. No, I think it's a good comment. And before I leave the word to Justin to sort of give another perspective on metabolism or metabolic theory, I'd like to ask all the participants, if they want to ask questions, to please type them or raise their hand. So Justin?
Yeah, one of the things that really is striking to me, and I think deserves a lot of attention, is how this idea of redundancy in systems, and how organisms can adapt to fulfill similar functions as others as they disappear or are replaced, etc. An example of this is fruit dispersers in South America. A lot of larger fruit was dispersed by larger mammals, for example, like the gomphotheres in the avocado example. And then when those organisms disappeared, the dispersal of those fruits was replaced by humans, and then later by domesticated animals. And so we have like these interaction niches that change over time and are dynamic over time, even within the time scale of a species, without including evolution. These ecological plasticity that isn't really realized until the community itself changes. I think understanding how these large diverse systems operate in the face of disturbance really is, to some extent, a product of to what extent there can be redundancy that may only be realized, post disturbance. And so I think, our understanding of the dimensionality of interactions is often very simple. We describe interactions in 1 or 2 dimensions, and in reality, the interactions themselves can be very diverse in their effects, and change over time in response to how the system changes.
Great. Yes, this point about redundancy, I think it's sort of connected, in some sense, an alternative. At least I see it like that, but perhaps I'm wrong, with what Mercedes is saying about the high-dimensional functional space and high-dimensional trait space, right? So if there is redundancy, it means that somehow the trait space, the functional space, is low-dimensional, and there are multiple ways to realize a given function. So I don't know if anyone wants to comment on this dichotomy, whether it is a dichotomy or...
I think there's a general question here also, which is how much complexity at the sort of phenotypic level and the interactions are needed to get a very diverse community. And that one is not, I think, is not at all clear. I think it's, for example, can it be that a rather small number of chemicals associated with the metabolism are really already sufficient to get you a high diversity? Or simple features of pathogen host interaction, is that already enough? So I think that's not clear. I think the issue about somehow the redundancy, it seems to be one part where this is absolutely crucial, but which is connected to all this, is in the evolutionary level, the fact that there are very many different ways that an organism can do somewhat better in a given environment, right? And then for any ways it's going to change its sort of organismic phenotype, there are very many changes that it can do, the possible genetic changes, and then sort of at the nanophenotype level that can give rise to that, or to give rise to something close to that. And that may end up sort of having consequences that there are always going to be different ways of replacing certain of the functions of that sort of redundancy. But I think that's another one of those sort of, big number things that we don't understand at all, that it tends to get way underemphasized. And certainly game theory, unfortunately, does this, is that there are extremely large number of possible strategies in a loose sense, right? And that's a really important important part of the evolution and presumably of the ecology.
Yes. Anyone wants to comment on this or other parts?
I guess maybe one comment is that I think there is really, this idea that functions rather than taxa ultimately dominate the community dynamics. I think, to me, is really fascinating, and it is in the end about redundancy, because genomes may have this mosaics of functions, and how exactly these functions are coupled in different genomes is an outcome of horizontal gene transfer, long-term evolution, and so on. So understanding whether there are aspects of this that have been optimized throughout evolution, I think, is very interesting. And yeah, I'll stop here. I think this is largely unexplored, and still.
Well, I think there is an experimental question there, which I suggested to my colleague Michael Fischbach, who works on gut microbes, and has these consortia of 60 gut microbes in the lab, which he claims fill up all the niches. Now, my other colleague Alfred Sporman says there's no such thing as bacterial niches, and one can't think of it that way. But I asked Michael, I said, can you do an experiment where you take one of the Bacteroides and put a lot of the other functions into it? So you move a lot of the operons that are supposed to be crucial of the niches filled by the other ones. And is it possible that one organism can at least, roughly fulfill many of those functions all at once? And then there's the immediate question: is if you then let the system evolve, does it immediately diversify into a whole bunch of organisms that do them better, each do them better? So I think there are some things that one could really try to actually do experimentally on that in moderately complex complex communities.
I think one of the other important things to consider is just the importance of the constraints in the system. Consider the Rubisco protein and how little it's changed over so much time. You think, really, you can't think, there isn't something better that could evolve, but it's almost this evolutionary kind of dead end. Obviously, it's a very successful protein in doing what it does, but organisms have found incredibly complex ways to get around the constraints set by carbon fixation. Designing entire physiologies to reconstruct early atmospheres to be more efficient within their tissues. And, I think a lot of diversity, stems from the limitations in the system, just having strong constraints and then many pathways around those constraints that different types of organisms have to follow. And that might not have been the case if there was always a better, if there wasn't a constraint and everything was following the same pathway towards higher and higher fitness peak.
And then each evolutionary accident as to which direction is taken constrains and opens up possibilities for all the future directions. Right?
Yeah. So anyone else wants to add something on this? Or someone from the participants, if they want to ask a question, since we have 10 minutes toward the end. When nobody wants to ask anything. So I mean, one sort of direction that has been sort of a little bit explored in this discussion, but perhaps can be unpacked a little bit more, is a question that actually emerged during one of the lectures, which is exactly related to evolution and evolution of diverse communities. So what are the conditions under which evolution produced these highly diverse communities? So we said these trade-offs and constraints. So I don't know if anyone wants to comment on that and add something.
I'll say something. I think this is actually a case where Mercedes' original point that there are some useful abstractions from the two species case to end speciesisms could be useful, because if there is going to be some diversification, it's going to be one splitting into two in a simplistic sense of the word. I know it's more complicated. And presumably those two do interact more intensely than they than they do with anybody else in that system. And I think it's very interesting in the sense that we've been very good at trying to think about this problem in terms of some sort of stabilization, regulation of the coexistence that emerges from those two species that form. But we've really not paid much attention to what comes out of some very simple two-species coexistence work, which is that there is probably some competitive imbalance as well. And something about the need to think about that competitive imbalance axis at the same time as we think about the stabilizing niche differentiation, if you want to call it that, axis is probably worthwhile and hasn't been done. And you probably wouldn't have thought of that had you not gone through a theoretical ecology experience at some point in your career, if that makes sense. So interestingly, people have been so good at focusing on one axis of the problem and not really thinking about the other. And I think that really highlights a real gap in what we are approaching on this problem right now, theoretically and empirically.
Yes.
Yeah, I think I mean I agree, and I think one of the reasons I sort of want to focus on strains, or two species with lots of interacting strains. It's sort of the beginning of the diversification, and one hopes that one doesn't need all the ways in which many complex species interact with each other if one understands more some of the basic ones in which two species do, and then what can happen within them. And in some ways, it's learning to walk before one tries to run. And I think my sense is we're still at a stage for understanding diversity and so on of really crawling, right? I mean, we're trying to get up on our feet and sort of go some stumble steps forward. And I think there's where there's luck and experience and various other things of sort of choosing systems and models and so on, which might make one go forward. I think one of the really nice things from this school is it's become clear the sort of diversity of ways of approaching these kind of problems, even from people with relatively similar backgrounds, at least superficially, right? And of course all the things from different from different backgrounds. So I think these exchanges, if everyone agreed on what was important, it would be totally boring, and we would almost certainly all be wrong.
Well, I totally agree. Yes, Mercedes, you want to say something?
Well, I think Daniel touched on an interesting question because we either heard about species or we heard about strains because that's convenient, and we are kind of avoiding the sort of macroevolutionary question in a sense of how do you connect the two, and of course that is very interesting because even in the world of strains, what also evolves is the reproductive isolation. In systems with incredible recombination, for example, there are groupings of genes, for example, that recombine more with each other, and some that are more conserved, and so on. So this building of the barriers, even within, for example, pathogen strains that reassort and recombine, it's not like a free-for-all. And this, we see this even at that level, the buildup of these barriers, which I think is a very important part of this evolutionary process. And I think it's another area we don't fully understand. And it's, how do we go also, of course, it's the old question going from the microevolution to the macroevolution, that I think is a fascinating question because I don't think it's so discontinuous. I mean, you also see it within strains. Sorry.
Yes, Justin, you want to say something about these sort of macroevolutionary aspect and deep time changes?
Yeah. Well, I guess I would just argue that that a lot of the energetic constraints, a lot of the kind of simplistic perspectives that we put into how we capture how interactions shape systems, they're at least you might be able to argue that they're better when evaluating macroevolutionary systems because you're really looking at these big general trends and you've zoomed far, far away from kind of the noise of ecological systems. And I kind of coming from a deep time perspective, I always am concerned about how much we're focusing on contemporary ecosystems, much, much more than we tend to look at kind of these average quantities of systems that the fossil record averages out for us. And so in some sense, kind of the fossil record and the deep time perspective, it's already isolating out some of the biggest signals. And to what extent are we trying to apply these very simplified models to noisier systems that are much more difficult to try to understand? It's kind of like trying to predict where a molecule of gas is with, in the average gas law. It's impossible. But we do find, I think, if we try to integrate more of a deep time perspective and take advantage of systems that have been around for a long time, and the average signals that the signals that they leave behind, which have to be averaged, we might be able to leverage kind of ecological theory in different ways.
Thanks a lot, Justin. Anyone wants to add?
Maybe I'll add.
Yeah.
I'll add something. I think this is a macroecological perspective of a very different kind, but one of the things that I like to think of is you can view microbial metabolism as a planetary phenomenon, of course, and now from a very different perspective. That sounds like science fiction, but people look at the atmospheres of extrasolar planets, right, in search for signs of life, and there is a lot of interesting research on non-equilibrium of chemical systems on these extrasolar planets, and I think that it's really interesting to think how the overall chemistry at such large scale, with just an influx of energy from light, and given the molecules that are present, how does that link to the scale of microbial metabolism we were used to think of, and the microbial ecology side of this? And I think there is also room for what I think is really interesting way of thinking about this in terms of just, besides the details of the chemistry, in terms of the flow of electrons that are kicked up by solar energy and gradually find their way down to lower energy levels. And I think there is a lot of potential for linking this large-scale theories of non-equilibrium biospheres to the lower-level microbial metabolism models. I know this is a little bit out there, but I think it's something interesting.
Yeah. Great. So we have less than 1 minute. So anyone wants to give some final thoughts? Or any question from the participant?
I just have one comment. I think is one of the things which often doesn't get into papers is really sort of struggle for the good question. And certainly doesn't get into proposals I've discovered, or at least they get rejected if they do have those things. And somehow the writing sort of more things down about not just, okay, this is the question. This is my sort of struggling to try to make good questions out of these very things. And when things normalize a bit, I've been meaning for a long time to get a series of talks going called Bio-Y, something called Bio-X at Stanford. They're called Bio-Y, with a letter Y, but where it would really mean the WHY, and really trying to get people to talk about questions that they didn't even quite know what the question was yet. And I think this discussion has been really good that way. But it'd be really nice to if people, the community generally, took effort to write such things, write things down, maybe collectively. It'd be fun.
Yeah. Mercedes, you wanted to say something, or you're muted?
Oh, no, I was listening for,
okay.
for a while.
Okay. Well, there is actually one question from a participant, Monday.
Thank you so much, the panelists, for giving us the roundtable talk. Please, I want to get a real take-home message regarding this intuition and modeling. Is it that when you want to model, you need to first of all have your intuition on what exactly you want to get, or is it the other way around? Please, I need a key take-home message so that I can get something and get it clearly from here. Thank you.
Anyone wants to respond? Thanks very much for the question.
I can only speak for myself, but when I start designing a modeling approach, I usually think I know what I want to get, and that often isn't what I get, and it changes my understanding of what I was actually wanting in the first place. So for me anyway, it's a method of kind of clarifying what I know and what I don't know, and it usually doesn't. What I think what I want to know when I begin doesn't match up exactly where I end up, but that's just me.
Yeah. Anyone wants to add something or ask a question? Great. So if not, I want to thank Stefano, Daniel, Jonathan, Matteo, Mercedes, Daniel, Segre, and Justin for this very nice discussion. I think we could go on with this discussion for other 50 hours. And in fact, we, with Mercedes and Matteo, we have decided to have a follow-up of this school, which will be a workshop to be held again online in a month from now, so we can sort of take a break from Zoom and screens. And I think all the participants will receive information about that. And there will be a lot of space in that workshop to discuss about diversity and the limits to diversity assembly. So with that, I again, thanks to all the panelists. I also want to thank all the participants. There are now 60 people connected to this Zoom, and looking at the names, I saw that most of the people actually attended like all the 52 sessions. So that was pretty impressive, and I really appreciated that. I mean, also the interaction with you was great. I'd like to thank, beyond the lecturers of this session, all the people that lectures and gave talk during this school. Their contribution was great, and I think there was a lot of material that has been put forward that could be available for many people that wants to enter in this field or understand what are the challenges and the opportunities for research. I'd like also to thank the people in the sort of in the background that you probably have not seen. I'd like to thank Monica, Victoria, and Adriana, the secretaries of the school, and in particular, I'd like to thank Massimo Maffione, who gave the IT support and really stayed here for 52 sessions. So, and at the end, I'd like to thank Matteo, Simon Levin, and Antonio for organizing this with me. So this is the end. Thank you very much. Now stay away from Zoom for a while, and I hope to see you either virtually or hopefully physically here in Trieste soon. So thank you very much.
Thank you, Jacopo.
Thank you very much.
Yes, next time in Trieste, because
yeah, yes.
That would be a much more interesting discussion.
Yes, now, yes.
We know from experience. We know
Yes.
from